Hi everyone. Good evening, everyone. Good evening to all. So I am so excited to start with this unit-wise final revision. And before that, I was reading your chat. Thank you so much for such a warm welcome. And without a further ado, we'll get on to our final revision today. So before we start with this series, so we have 10 units. We'll take 10 days to complete all the 10 units. And unit-wise revision. In this unit-wise revision, see, first of all, you need to understand what's going to be the strategy for this revision. We have to recover and we have to rehaul all those points that we might be missing during our revision. So we keep on reading our NCERT, non-stop, but we tend to miss out on the keywords. So now in these classes, I am going to bring those keywords all together. We'll be revising everything in detail, everything you will see. Just the difference is I have got these prepared notes. So I you will be getting the PDFs also. Take down those notes, revise them, and while I am teaching, keep rethinking that whether you remember it or you not. In case there is any doubt, please feel free to ask. Voice should be audible. See, voice is absolutely audible. Voice should be audible. How long is this session? I don't know. At least till 8 o'clock. Before, we are not going to stop till our 4 chapters from unit 1 are done. Okay? So we are not going to stop till these 4 chapters are done. So now I will recommend all of you to quickly take your copies with me. Bring your copies, take a pen, keep your NCERT in front of you or your already made notes with you. So keep these things and keep revising with me. You don't necessarily have to make the notes all over again. Okay, but make sure that you have not missed on any keyword. So if you have not highlighted anything important, do that with me. If you want to write down the keywords with me again, that would be fantastic. Okay, PYQ solving will also be done. Yes, I'm going to teach everything, botany as well as zoology. We're going to take it. So shall we start? Give me a quick thumbs up, all of you. Everybody give me a quick thumbs up that you're ready with your copies, you're ready with your pen, and you're ready with your NCERT. Quickly, I want your thumbs up, and then we'll start without any further delay. Yes, you can score 340. You can score 360 out of 360 from these sessions because I'm not going to miss out anything. And I'm also going to discuss few questions, very important questions in every session. Okay, all ready? Sessions time would be 5 to 7:30 or 7:45, may go up to 8. But we'll try to finish it off by 7:45. See, time is also a constraint, right? So we are going to practice it now. So I need your 100% attention now, and let's get started. Right, absolutely. Can we write notes with you? Yes, you can. See, I have brought just keywords. Okay, and you also have to write down the keywords. Now this is recovering everything that has gone in our subconscious mind. Okay, let's get started. Cool. So this is unit-wise final revision. I'm sure you have attended so many one-shots, chapter-wise one-shots. Now we are going to revise this unit-wise. Okay, the 4 chapters we are going to discuss today is living world, biological classification, plant kingdom, and animal kingdom. I am going to start with biological classification. Living world, why I'm not starting? Because living world is not very important chapter. We hardly get 1 or 2 questions, not even 2. Last year we didn't get any question from living world, but as always, NT is full of surprises. We are not going to leave any chapter, but I thought we'll start with biological classification. So these are the keywords. You will be making these notes with me. That would be fantastic. Okay. All right. So biological classification. This is chapter number 2 of your NCERT. And yes, here it is. So out of biological classification, we know that the 5 kingdom classification that we go by is given by R.H. Whittaker. That was given by R.H. Whittaker, right? And according to R.H. Whittaker, let me change this. Okay. R.H. Whitaker gave us 5 kingdoms. What were those? Monera, Protista, Fungi, Plantae, and Animalia. Now, the most important point here to be remembered is what was the basis of the classification. See, in every classification, you must remember what was the basis. So, in point 1, the basis on which he classified all the animals into 5 kingdoms was cell structure. Second was thallus organization. You can also call it body organization. Third was mode of nutrition. Fourth was reproduction. Five was phylogenetic relationships. Then next it was whether the cell type, whether the cell was prokaryotic or eukaryotic, right? That was the cell type. Clear? And can anyone tell me what was one more basis of classification? Anyone? Quickly let me know. So one was cell type, whether it's eukaryotic or prokaryotic. Cell structure, whether the organism is unicellular or multicellular. Thallus organization, in which we see whether it is cellular level of organization, or the body is made up of tissues, or it is made up of organs, or organs will be classified, will be further forming the organ system, right? And then mode of organization, whether the organisms are autotrophic or heterotrophic, all these. Morphology. Thank you so much. Thank you so much, all of you. Thank you so much. All right. Then reproduction and phylogenetic relationships. So what are phylogenetic relationships? Phylogenetic relationship means the evolutionary history. So R.H. Whitaker considered all these factors while classifying the organisms. Is that clear? Perfect. Nuclear membrane, yes. Nuclear membrane, whether it is present or not, on that basis only, Vishwa, we classify the cell into prokaryotic or eukaryotic type. Are these points clear? Then next we have, now we are going to take every kingdom one by one, starting with kingdom Monera. Starting with kingdom Monera. Now, see, we know that kingdom Monera consists of only prokaryotic organisms, and out of these 5 kingdoms, it is only kingdom Monera which has prokaryotic organism. Rest all the 4 kingdoms include eukaryotes. Are these points clear too? Are these points clear too? Perfect. Then prokaryotic, being prokaryotic means it will not have nuclear membrane, it will not have any membrane bound organelles, no mitochondria, no ER, no Golgi body, right? The only organelles which are present, quickly let me know in the chat, the only organelles which are present in prokaryotes are which? Which are the only organelles present? Quickly let me know in the chat. All are unicellular. Cell wall will be present, and this cell wall, usually cell wall is present, and this cell wall is non-cellulosic, means it is not made up of cellulose, rather it is made up of this peptidoglycan. Very important name. It is made up of peptidoglycan, also known as murine. Are these points clear? Then see, we know that Monera includes, what does Monera include? The kingdom Monera includes your bacteria, the cyanobacteria, which is known as blue-green algae, right? It also includes mycoplasma, yes or no? And it also includes PPLO. These are the things. And bacteria can be of 2 types, eubacteria and archaebacteria, right? We'll be talking about them one by one. Perfect. The only organelles which are found in these organisms are ribosomes. And that too also 70S type. Perfect. All right, moving on. So now these bacteria which are motile, they will have flagella. So flagella may be present, flagella may not be present. It may be positive or it may be negative. It may be present or not present. If they are present, such bacteria will be motile. If they are not present, they will be immotile. So flagella is always a motility providing structure. Then the organisms are going to divide by binary fission, and this represents the asexual mode of reproduction. This is a asexual mode of reproduction. And then recombination, conjugation, genetic recombination can occur through conjugation, transformation and transduction. This represents the sexual type of reproduction. Please let me know, are these all points clear to you? And now when you are reading these points, everything is coming back. See, it's very important that we are able to recollect what we have learned in the entire 2 years, Recollection is very important, and recollection comes from revision, right? MCQ battles will be done. Definitely, I'll be taking the MCQs in this session also. I'll be taking MCQs in the free, like special classes also on app. Okay, so questions will be discussed. Don't worry about the questions. Perfect. Now we know that kingdom Monera was further divided into two categories. So bacteria we have of two categories. One is eubacteria, second is archaebacteria. Let's see what is the difference between the two. So the archaebacteria and this archaebacteria, wait. Okay. Now, archaebacteria. What do we understand by archaebacteria? Archaebacteria means primitive bacteria. These are primitive bacteria, and they are found in the extreme type of habitats, right? Now, what are the extreme habitats? Extreme habitats could be very high temperature or very salty conditions or purely anaerobic conditions. So these can be extreme habitats, right? So according to the extreme habitats, we have three different types of archaebacteria. One is methanogen, second is halophiles, and the third one is going to be thermoacidophils. The third one would be thermoacidophils. Okay, perfect. Now, what is that one important property of archaebacteria which allows it to survive in these extreme habitat? So that property is a difference in their cell wall structure. This different cell wall structure. So here we have assertion and reason. So see, we are going to discuss assertion and reason side by side. So they are able to survive the extreme habitats because of different cell wall structure. Now, what is the different cell wall structure like? The lipids present in their membrane, they are very unique and they are not found in eubacteria. Are these points clear? Ma'am, please teach by writing on the board. Oh, I would love to do that. But right now, let me explain you one very small thing. Please cooperate with me. I am right now at a very remote place, okay, where I don't have my studio set up yet. So when Unacademy said that, okay, I have to take classes, I said, okay, let's quickly start with the classes. Very soon, I'll be shifting to a better place where I will have my studio set up and we'll be studying on the board. So let's cooperate. Let nothing stop us, okay, from revision. So no lack of energy and not getting. Yes, please. Ma'am, please. Okay, no problem. Thank you so much. Thank you so much. All right. So the three types of archaebacteria are methanogens, we have halophiles, and third would be thermoacidophils. Now the name indicates itself what it's going to be like. Methanogen. See, I have always believed and I have felt that in biology, since there are so many terms, the only thing that makes bio easy is if you learn how to break the term, right? This is called word root. Now, methanogen, it already says that they will be methane producing. So these are found in marshy areas. They produce methane, and where else it is found? In the rumen of the cattle. So rumen is a special chamber in the stomach of these cattle. So we call such organism as ruminants, right? Ruminants. These bacteria help in cellulose digestion. These help in cellulose digestion in case of ruminants. Clear? And then also they are found in biogas plants. So they help in biogas production also. And as we understand, the major gas present in biogas is what? It is methane. Okay. So the bacteria which are responsible for methane production are archaebacteria. And which archaebacteria? Methanogens. Is this point clear? Perfect. The second type that we have is halophiles. So as the name says, halo word is for salt. Philes means loving. Halophiles are found in extreme salt conditions. And the third type is thermoacidophils. The name itself says they'll be found in hot conditions, hot springs, hot water springs. Acidophils, they'll be also able to survive in the acidic conditions. So such bacteria are called thermoacidophils. Are you clear with that? Can anyone tell me in the chat an example of thermoacidophil that you have learned in your syllabus? Anyone? Example of thermoacidophil that you have learned in your syllabus? Anyone? Okay, I'm giving you a hint. It is in biotechnology. It's a very important bacteria. Quickly let me know which is that bacteria that you have learned. Very good. The first one to answer is Maam. It is Thermus aquaticus. Remember the source of Taq polymerase, that is thermoacidophile. Perfect. It gives us Taq. You know? All right. So that was the first type of bacteria, which is arc bacteria. The second type of bacteria that we are going to talk about are eubacteria. Now, U means true. These are true bacteria. They have rigid cell wall, which will be made up of peptidoglycan. This will be made up of peptidoglycan, not cellulose. Let's revise again. And in case they are motile, they can move, then they must have flagella. Okay. Now, eubacteria are further of different types. The first type of eubacteria that we are going to talk about is cyanobacteria. Okay, the cyanobacteria, blue, also known as blue-green algae. Cyanobacteria is also known as blue-green algae. Clear? Now, these blue-green algae, they have pigment, the same pigment which is found in plants. It is chlorophyll A. That is why this is also known as universal chlorophyll. Any organism which is photosynthetic will have chlorophyll A, and so does these blue-green algae also have. Is this clear? Perfect. So they are photosynthetic. They can make their own food. So they are autotrophs. Apart from chlorophyll A, they also have phycocyanin. They also have carotenoids. Now they are unicellular. They can live in colonies or they can be filamentous. See, these are all keywords. I have told you we are going to revise the keywords written in NCERT. No more full sentences, just the keywords, which should always strike back. Where can I get the PDF? You will get the PDF, Jimin, at the end of the session. They will be attached in the description box. You can easily find the notes. Okay. Yes, full schedule of the revision program. Classes will be alternate 5 p.m. to 8 p.m. And in every class, we will be discussing one complete unit. Okay. Rice fuel, I hope the things are clear. Okay. All right. So they can be unicellular because, of course, Monera, being moneran, it has to be unicellular, it has to be prokaryotic. Some of them can be colonial and some of them can be filamentous. Clear? And all of them, they have a mucilaginous sheath. Can anyone name a cyanobacteria for me? Anyone? Before I tell you the examples, can anyone name a cyanobacteria? Yes? Nostoc. Perfect. Then Anabina. Now both of these, they are involved in nitrogen fixation. So can we call them biofertilizers? Yes or no? Can we call them biofertilizers? Right? Okay. Now this nostoc and Anabina, see, I'll be revising all the diagrams at the end of the session. All the diagrams from this one unit will be revised at the end of the session. Now see, now this is a filamentous and colonial nostoc. Clear? Now there is present, as said, a mucilaginous covering. There's a mucilaginous covering around the whole body. Okay. So this is the mucilaginous covering which protects it from dehydration. Okay. So this is the mucilaginous covering which protects it from dehydration. Now, if you have read your chapter Microbes in Human Welfare very carefully, you must remember it was written that cyanobacteria are used. They are used in paddy fields. Do you remember that line? How many of you remember that line? See, we are going to remember. We are going to revise the whole syllabus. We are going to revise the whole syllabus and you must know how to connect the dots. See, that becomes a huge weakness. When we get one statement based question, there are some ways in which NTA asks this question. It will connect different chapters, and that is where we flip. So you must be able to recall different chapters at different times. Okay. So these nostoc and Anabina, cyanobacteria is an important biofertilizer in the paddy fields. But we don't really use these cyanobacteria in the paddy fields. Why not? In actuality, see, theoretically it is possible, but in reality, we don't really use cyanobacteria much in the paddy fields because of this mucilaginous covering. It makes it slippery. The paddy fields will be super slippery. That is why they are not recommended. Okay. Then also, now see, this is a colony. This is a colony of nostoc. This is in the filamentous form. Now, there are certain cells. These cells, will be known as heterocysts. These are specialized cells which are going to be involved in nitrogen fixation. Now, the whole Anabina is not involved in nitrogen fixation. Only certain cells will be involved in nitrogen fixation, and these cells are known as heterocysts. Heterocysts are involved in nitrogen fixation. Why only these cells? Because these cells have an enzyme called nitrogenase. Are these points clear? So we need a nitrogenase for nitrogen fixation, and only these heterocysts have nitrogenase. Are these points clear to all? Everyone? Clear? All right. Now coming on to the examples, see, we have already revised the examples here. So the examples were nostoc and Anabina. Clear? Moving on to the next bacteria, which would be chemosynthetic bacteria. Right? So chemoautotrophic bacteria. Clear? So what are these chemoautotrophic bacteria like? So chemoautotroph, they are going to make their own food, that is for sure. But, how are they going to make their own food? They're going to oxidize the inorganic substances like ammonia, nitrites, and nitrates. So this is how they make their own food, by oxidizing the inorganic substances. So they derive that energy to make food from oxidation of these substances. For example, sulfur, iron bacteria, sulfur and iron bacteria, these are all chemoautotrophic bacteria. Okay, now then they release energy and ATP will be produced. And very important, what is the economic importance of these chemoautotrophs? Now, just as we have seen that cyanobacteria are important biofertilizers, cyanobacteria are important biofertilizers, the economic importance of chemoautotrophs are they help in recycling the nutrients. They help in recycling of nutrients. Mineral recycling. This is what NCERT has written. They are involved in mineral recycling. Are these points clear to everyone? Perfect. So see, when you are revising these topics with me, you must be able to recollect what is exactly written in NCERT. Clear? We have read NCERT multiple times. We remember what is written everywhere. But now the point is, when we revise, we must only click to the keywords. That is why I have made these point-wise notes for you. I have brought flowcharts for you. I'm not bringing NCERT here. NCERT also we'll be reading. See, there is another series that I am starting every day. I am going to send you very important lines of NCERT. It will be a 10 or 15-minute session. From tomorrow or day after tomorrow, you will get that, and you must revise. See, I'll tell you how you are going to revise things. Let's proceed. And during the session, we are also going to talk about our strategy during these last 15-20 days. Okay. Coming on to the third type, which is heterotrophic bacteria. Now, these are the most abundant bacteria. Out of all, these are the most abundant bacteria. Not blue-green algae, not chemoautotrophic. Who are the most abundant? It is heterotrophic bacteria. They can be saprophytic, they can be parasitic, they can be symbiotic. Can anyone name these for me? Any example of symbiotic bacteria? Yes, please. Any example of symbiotic bacteria? I'll give you a hint. It lives in the roots of legumes, root nodules of legumes, and helps in nitrogen fixation. Anybody? Absolutely right. It is rhizobium. Perfect. It is rhizobium. So you have also talked about a biofertilizer. See, this is also a biofertilizer. Of course it is. Anything which is increasing the soil fertility is a biofertilizer. Oh, what am I writing? Biofertilizer. But nostoc and Anabina are which kind of biofertilizers? They are cyanobacteria, right? This one is a heterotrophic cyanobacteria, rhizobium. Again, this also has nitrogenase. For nitrogen fixation, you absolutely need this enzyme. Clear? Then coming on to uses. So they are decomposers because you have written that they are saprotrophs. So saprotrophs means they will be decomposers. They will help in nitrogen fixation. Just told you rhizobium. Curd formation. Remember, lactic acid bacteria, lactobacillus, that is curd, that is also heterotrophic bacteria. Antibiotic production, pathogens also means they will be disease causing as well. So all those bacteria that you have read in the chapter of health and diseases and are disease causing, they are all pathogens and they are heterotrophic. Are these points clear? Penicillin, no, Aditya, we've made a huge mistake. Penicillin is a fungus produced antibiotic, right? Okay. Break? No, no, we can't afford a break, Yasir. We can't afford a break. Now, the okay, now next category of organisms which are found amongst the kingdom Monera are mycoplasma. See, about mycoplasma, what all you must remember, it is the smallest living organism. Can anyone tell me the size of the mycoplasma in the chat? Anyone? Yes. See, it is going to be a question round also, so that you also know what you don't remember. And I'll give you a hint. While I'm asking you questions or while we are revising this, the points that you don't remember, that don't which don't click you instantly, make sure that you are writing down those points, or you're just you're making those points as headings. Keep noting down those topics so that you know what to revise again and again. See, last few days, there are absolutely certain things that you tend to forget. Make sure that you're making those short notes, because the final day will be the day when you will revise only those points. Absolutely. Good evening. So, the size of the mycoplasma is zero point zero 0.01 micron meter or zero point 0.1 micron meter, zero point 0.3 micron meter. Yes, quickly. So, that is what you have to write down. The size of the mycoplasma. This must go in your doubt session. See, I have already compiled all the sizes together. So I'm going to release them as one small topic or session. So make sure that you do attend that. Okay, 0.3, right? 0.1 to 0.3 micron meter. This is the answer. This is the smallest living cell. It doesn't have a cell wall. Now, since it doesn't have a cell wall, so the size, so the shape is not fixed. Shape is not fixed. Now, the organism which doesn't have a fixed shape is known as pleomorphic. So, when the shape is not fixed, this is known as pleomorphic. They can survive without oxygen. That is, they are anaerobic and they are pathogenic. So, four things that you must remember about mycoplasma are, what are the four things that you must remember about mycoplasma? It is the smallest living cell. Virus is smaller, but it is not called living, right? So the smallest living cell would be mycoplasma. They are all pathogenic, no good mycoplasma. Then they are anaerobic. They can survive without oxygen. They don't have cell wall, so no cell wall. These are the four things you must remember about mycoplasma. Are these points clear? Okay. Now moving on to the next kingdom, which is kingdom Protista. Now, what are the general features about Protista? Now, after Monera, all the four kingdoms, all the four kingdoms will be prokaryotic. Now, these are also prokaryotic. I made a mistake. So, apart from Monera, all the four kingdoms are eukaryotic, right? Now, this is the first eukaryotic, which is unicellular. So, Monera is prokaryotic and unicellular. Protista is eukaryotic and unicellular. Most of them are aquatic. They can be photosynthetic or heterotrophic. They might have flagella or they may not have, and some of them will have cilia. Apart from this, then there will be some Protista which will not have flagella or cilia, rather they have pseudopodia. Just think of amoeba, right? All these are the locomotory structures. Now remember, we are actually going to classify them, okay? Now, they will show sexual as well as asexual reproduction. Coming on to the first type of Protista. See, we have, so this is Protista. So Protista and Protista will be of five types. Chrysophytes, we'll have dinoflagellates, right? Then we have protozoa, okay? Then slime molds, and the fifth one, euglenoids. Now see, This is one such kingdom which brings all the kingdoms together. Okay, this is one kingdom that which brings all other kingdoms together. How? See, I'll be revising how these five kingdoms were created. See, chrysophytes are plant-like. Dinoflagellates will also be plant-like. Why? How? We'll talk about it. Protozoans are animal-like. Slime molds are fungi-like. So the name, that is why slime molds, fungi-like. Molds are always fungi, but slime mold is not a fungi, remember. And then euglenoids. Euglenoids are both plant-like and animal-like. In fact, they are the connecting links. These are the connecting links between plants and animals. Are these all points clear? So that is why we say the kingdom Protista brings all the other kingdoms together. Are these points clear? Please let me know. Are these points clear to all? Perfect. See, don't forget to share this session with everyone. That is very, very important. Okay. I would love to see all the students attending these sessions and nobody missing these revisions. Okay. Clear. Notes will be available. Notes will be available. But the best thing would be that you write down these key points with your own hand. See, do you remember? In we used to practice writing things so many times. We develop muscle memory. So last time, this is the last revision. So start writing these points so that you don't forget them. Okay. Now, coming on to the first type of Protista is the chrysophytes. In chrysophytes, we further have two different types. One is diatoms and second is desmids. See, diatoms and desmids. Desmids are known as golden algae. Okay. Now, these chrysophytes will be found in fresh water as well as in marine water. They are photosynthetic. So, this is plant-like character, isn't it? Plants have chlorophyll. Plants are able to produce their food. Then they have cell wall. Again, plant-like feature. Yes or no? Now, the cell wall is made up of silica. Now, see, this is the classification, right? Now, out of all these different classes, chrysophyte has a cell wall. This has a cell wall. Euglenoids do not have cell wall. Dinoflagellates will also have a cell wall. Then slime molds will not really have cell wall. Only their spores will have cell wall absent. These will also not have cell wall. Okay, so cell wall will be present in chrysophytes, in dinoflagellates and in slime molds. Only spores will have cell wall, true cell wall. But otherwise, the cell wall will be absent. Are these points clear? I'll be talking about fungus, which will have sexual spores. We'll talk about those one by one. Let's go one by one. Okay. Now, the difference is that all of them which have cell wall is made up of different things. In chrysophytes, the cell wall has silica. Okay. The chrysophytes will have silica. In dinoflagellates, the cell wall will be made up of cellulose. Okay. Now, protozoans will not have cell wall and euglenoids will also not have cell wall. But instead of cell wall, euglenoids will have a proteinaceous covering, which is known as pellicle. This is made up of protein. While cellulose is a sugar, silica is silica. Are these points clear? Now, coming back. So, when we were talking about, see, we were talking about the silica in the cell wall. This siliceous cell wall will make it indestructible. So what's going to happen? Even if the organism dies, even if the chrysophytes die, the cell wall is going to remain behind. This cell wall is indestructible. Now, this indestructible cell wall, what is going to happen here? This indestructible cell wall, when the organism dies and the cell walls remain behind, the earth which has the remnants of these cell wall of silica, that earth will be known as diatomaceous earth. And what is the function of diatomaceous earth? This earth is used, this soil which contains the remnants of diatoms, this soil can be used in filtration and polishing. Filtration of oils and syrups. Remember, this is what NCERT says. Second very important point about chrysophytes is that the cell wall is made up of two overlapping shells. Very, very important. See, this is such a unique character that you will not read in any other organism in the entire biology. What is that? The cell wall is made up of two overlapping shells, and these shells are going to fit into each other like a soap box. So few things that you should always remember when we are talking about chrysophytes. See, when we talk about chrysophytes, few features should always come to your mind. What are the unique features? Soap box like. This is the point you will only read about chrysophytes. Okay, and particularly diatoms. Then, since the cell wall has silica, they are going to form diatomaceous earth, right? And now this diatomaceous earth is also known as kieselguhr, which will be used for filtration and polishing, just like sandpaper, because it is of gritty type, right? We can use it. Are these points clear? Then coming on to the next type of protista is dinoflagellates. Now dinoflagellates are marine. They are also photosynthetic, but they have cellulosic plates. They have cellulosic plates. Are these points clear? Now there are many cellulosic plates. The whole body is covered by multiple. See, for example, this is a dinoflagellate, right? So these dinoflagellates, the body will be covered by these multiple cellulosic plates. This is how the body is present. Also, the name comes from dinoflagellates, means it has two flagella. Very important NCERT line. Is that clear? It has two flagella. Now see, the two flagella, out of the two flagella, one is longitudinal. This is the longitudinal, and the second one is the transverse. Is that clear? This is the transverse flagella. This is the longitudinal flagella. Are you getting all these points clear? Yes or no? Yes, one longitudinal, second is the transverse. So the transverse flagella goes through this transverse groove. Groove means a deep gap, right? It passes through the transverse groove, right? Now these dinoflagellates come in many colors. They have many pigments, so they come in various colors. It can be green, can be red, blue, brown, yellow. So there are multiple colors in which dinoflagellates are seen. Now there is one such dinoflagellate in the name of Goniolus. Now this Goniolus, which is a dinoflagellate, right, Goniolus, this is red in color. So when there's a tide containing Goniolus is rushing towards the shore, it looks like a red velvet. Okay, it looks like a red velvet coming towards the shore. So we call it red tide. So who causes red tide? It is the Goniolus. But everything that glitters is not gold. This red tide is not gold. This Goniolus is toxic. It is toxic. So the marine fishes, when they intake this water containing Goniolus, it is toxic for them. They die, right? So red tide is not good for them for the fishes. Are these points clear? Noctiluca, yes, Noctiluca is also there, Shri, but Noctiluca is blue in color, as blue as the markings here. It is blue in color. Okay. So we just have one example in NCERT, that is Goniolus. So we'll stick with Goniolus only. Now, another unique feature sorry, of these dinoflagellates, they are bioluminescence. They emit light. They emit light. So at night time, if you watch them, so they look very pretty because they are emitting light. And this property is known as bioluminescent. Are these points clear? Okay. Now the third type of protista that we are going to talk about is euglenoids. Now euglenoids are freshwater. Euglenoids are freshwater and they have flagella. Rather, they also have two flagella, but the two out of the two, one is very small. One is very small and one is long enough. So usually we get to see the longer one and the small one is really small. Secondly, I already told you, the euglenoids, they don't have cell wall, rather they have pellicle, and this pellicle is made up of protein, right? It is a protein rich pellicle. Now this pellicle makes it flexible. The cell wall is always hard, be it made up, no matter what it is made up of, silica or cellulose or peptidoglycan. Cell wall is always rigid in nature, but in euglenoids, cell wall is not present. So they have instead a protein rich covering known as pellicle. That is why the body is flexible. They don't have cell wall. Are these points clear? See, this is animal-like feature. Absence of cell wall is animal-like feature. Yes or no? But it does have pigments, the same pigments which are found in higher organisms like plants. So they have the same pigments, same pigments as in higher plants. Okay, and they will be photosynthetic, and being photosynthetic is the plant-like character. But it is only able to produce food. It is only able to photosynthesize in the presence of sunlight. So euglenoids have a very interesting nutrition. We call it mixotrophic. What do we call it? Mixotrophic nutrition. So in the presence of sunlight, if sunlight is there, it will be photosynthetic. If sunlight is absent, no sunlight absent, then it will be heterotrophic. Are you getting that? In the presence of sunlight, it is autotrophic or photosynthetic, but in the absence of sunlight, it is heterotrophic. And in heterotrophism, what does it do? It predates, it shows predation, it preys upon the smaller organism, it ingests them, just like us. So this predation is an animal-like feature. Yes or no? So now do you understand which are the animal-like features and which are the plant-like features in case of euglenoids? Yes or no? And the example is euglena. Example is euglena. So that is why we called euglena as a connecting link between plants and animals. Are these points clear to everyone? Are we able to recollect everything? How many of you are making notes with me? How many of you are writing down these points? Just a list of it. Quickly let me know. How many of you are making quick notes, just writing down the points along with me? Quick. Let me know in the chat. How many of you are doing that? Shri? Okay. Nobody else? Sonakshi? Cool. All of you are? So make sure you're writing down. You're building that. You're letting your hands also remember things now for the final go. Underlining it in the book. Okay, Vishwa. But I would still suggest, I'm sure that you have already underlined so many things in your NCERT. So how would you highlight them now? Okay. Coming on to the next type, we have slime molds. This is the fourth type of Protista that we are going to talk about. And this slime mold is fungus-like. This is fungus-like. Now, just like fungus, it is also saprophytic. Being saprophytic, it is going to feed on the dead organic matter. Now, slime molds, when the conditions are favorable, they would make an aggregate. In favorable conditions, they will just, like, when we are chilling out, we just sit around, we gossip. Now, similarly, in slime molds, when there are favorable conditions, they will aggregate. Now, this aggregation is known as plasmodium. So, in favorable conditions, they will make an aggregation, and the aggregation is known as plasmodium. See, do not confuse this term with the plasmodium, the malarial parasite. Okay, this is the name of a group. This is the name of their party. This is the name of their aggregation. And when do they form this aggregation? Only during the favorable conditions, right? But during the unfavorable conditions, as unfavorable conditions arrive, they start forming fruiting bodies. And what do fruiting bodies produce? They produce spores. Are you getting that? So in favorable conditions, plasmodium is the name of their aggregation. Okay. And during the unfavorable conditions, they start producing fruiting bodies. Okay. And these fruiting bodies will lead to the formation of spores. Now, what should you remember about the spores? Very important. These spores will have true cell walls, and they'll be highly resistant spores. And the third point, they are always dispersed by air currents or wind. So air will disperse it from one place to another. The three things you must remember about the spores of slime molds. Are these points clear? Please let me know. Keep giving me thumbs up that these points are clear. The three things you must remember about the spores of slime molds are: they have true cell wall, they're highly resistant, so they can discard all the, they can survive all the unfavorable conditions. And the third point is they are dispersed to other places through air currents. Are these clear? Perfect. Now the fifth and the last type of protista that we are going to talk about are protozoans. These are called protozoans. Wait. Yes. These are protozoans. So as the name itself says, proto means primitive, right? So protozoans, yeah. So protozoans. So I'm going to talk about the fifth type of protista, that is protozoans. So name itself says primitive animals. These are primitive animals. Now these protozoans, very important question again. These protozoans can be classified on the basis of, can anyone tell me that what is the base of their classification? What is the basis of this classification of protozoans? Anybody? What is the basis? See, basis of classification are very important no matter what are you classifying. Yes, please. So what is the basis of classification here? What is the basic? Yes, structure of locomotion. So we are going to classify them on the basis of structure of locomotions used by them. Some of them are going to use cilia, some of them are going to use flagella, while others will use pseudopodia. And then there will be fourth type which doesn't have any specific locomotory structure. And that is why we have four different types. The one which use cilia for their locomotion are known as ciliated protozoans. The one which use flagella will be known as flagellated protozoans, right? And those who will have pseudopodia will be known as amoeboid because it represents amoeba, right? And those which do not have any specific are known as sporozoans. They are known as sporozoans. Are these points clear? So on the basis of locomotory structures for locomotion, we will classify them. Are these points clear to everyone? Are these points clear to everyone? So we have ciliated, flagellated, we have amoeboid, sorry, we have amoeboid, and we also have sporozoans. Now, we have named them on the basis of their locomotory structures, but why do we call sporozoans as sporozoans? Because they will show a specific stage. They will show a specific stage known as sporozoids. This is one stage. Do you remember malarial cycle? Right? So malarial cycle, the plasmodium comes under sporozoan. I hope you remember the examples also. Anyone who doesn't remember the examples? Okay, let's see here. In ciliated, we need to remember one example, that is paramecium. We have others, but we'll only stick to NCERT. Paramecium. In flagella, we have two examples. One is trypanosoma and we have leishmania. Both are pathogenic. Remember, they both cause diseases. Pseudopodia using organisms are amoeboid. We have amoeba, we have entamoeba. Entamoeba is again pathogenic. It is disease causing. It causes amoebic dysentery, right? And then in sporozoans have sporozoid stage. But the example of sporozoans is plasmodium. Now this plasmodium is different from the plasmodium that we have talked about in slime molds. Are these points clear to everyone? Right? Then coming on to the next. Yes, please. So these, let's talk more about them. See, quickly read. So amoeboid have pseudopodia, right? They can be freshwater, marine or moist, and some of them are parasitic. For example, entamoeba. Clear? Now the marine ones, remember, marine ones will have silica around them. They have silica shells around them. Only the marine ones will have silica in their shells. Is that clear? Not everybody will have. Then, in flagellated, they are free living or they can be parasitic. They will have flagella. Example we have already done. Just recollect trypanosoma and leishmania. Cilia will have cilia, gullet present in paramecium. Coordinated movement will be because of cilia. So all the cilia are going to move in one direction, which will propel the organism forward, and it has holozoic mode of nutrition, so food is ingested. Clear? Then sporozoans, they have the special stage, sporozoid stage, which is infectious. Remember, the stage which really enters the human body before the malarial fever actually sets in, that is the sporozoid stage. So it is the infectious stage. Are these all points clear? Yes, nuclear dimorphism, you're very right. Paramecium does show nuclear dimorphism. It does. Okay, there is one small micronucleus and meganucleus, but again, NCERT doesn't tell us about that. So, examples are Plasmodium. Clear? Now, coming on to the third very important kingdom, which is kingdom fungi. Okay. Now, kingdom fungi, again, it consists of eukaryotic organisms. All of them are going to heterotrophic. Some of them can be parasitic. They can be saprotrophic or symbionts. Can anyone tell me an example of a symbiont fungi? Yes or no? It is mycorrhiza. Do you remember? Mycorrhiza. Myco word is for fungus, lives in the roots of higher plants, especially pinus. In plant kingdom, we talk about it. In parasites, we talk about the ringworm. Remember health and diseases? Ringworm. Ringworm is a fungal disease. It is parasitic in nature. Clear? And then saprophytes. All the saprophytes are decomposers. Then it has cell wall, and the cell wall will be made up of chitin. Now the body consists of thread-like structures. And these thread-like structures are known as hypha, and these hypha will form a network. The network will be known as mycelium. Now this mycelium can be septate or aseptate. See, this is a hypha. So, in case there are no cross walls present, I'll call it aseptate. Septum absent, aseptate. In case the cross walls are present, like this, so this will be known as septate. Are these points clear? It will form spores. Spores can be asexual as well as sexual, right? So it will show both types of reproduction. Are these all points clear to everyone? Now, one thing that I want all of you to write down separately, that is the name of the spores. Please make a separate list. So I'm going to talk about asexual spores here and sexual spores. See, very, very important point. And this is where we always confuse and we make mistakes. Now, this is the final revision and we are going to learn it for once and for all. Now see, we have four different classes in fungi. So first of all, we'll remember pad B. In this session, I'm going to tell you about the tricks also, so that now things just stay. Now see, the first of all, fungi will be divided into four classes. Also get ready for my next question. What is the basis of classification for fungi? Very important question, and it is a PYQ. But anyways, please focus here. So we have P for Phycomycetes, A for Ascomycetes, D for Deuteromycetes, and B for Basidiomycetes. In Phycomycetes, we know that asexual reproduction is present. In Asco also it is present. In Deuteromycetes also present. Basidiomycetes do not show asexual reproduction. Yes or no? Phycomycetes shows this. Deuteromycetes do not show sexual reproduction, and Basidiomycetes show sexual reproduction. Are these points clear to all? Yes or no? Please let me know. Now we are going to talk about the spores which will be formed. In Phycomycetes, in sexual reproduction, we have zygospore. In Ascomycetes, we'll have ascospore. Deuteromycetes do not show sexual reproduction, and Basidiomycetes will have basidiospore. Are these points clear to all? We'll come to toadstools. Wait a second. Okay, now talking about the asexual ones. In Phycomycetes, there will be two types of asexual spores, zoospores or eplanospores. If they are motile and they have flagella, they will be zoospores. Zoo means animal, animal-like feature. Motility is animal-like feature. Zoospores are motile. Eplanospores are non-motile. Are these points clear? Then in Ascomycetes, we have Cnidaria. Deuteromycetes also form Cnidaria. See, from this one table, three different things you are learning. First of all, you are learning that which class of fungi will show which kind of reproduction, sexual or asexual. Second, the type of spores which will be present, asexual and sexual. And then third thing that you're going to learn is very important, very important, that which of the spores are formed endogenously and exogenously. So let me tell you, these are both endogenously formed. These are both, wait a second. Endospores and planospores are endogenously formed. These are endogenous, while this is also endogenous. Ascospore is also endogenous, while basidiospore is exogenous, formed on the outer side, exogenous. Conidia is always exogenous. So this one table is going to revise everything for you. What is endogenous? Okay. So let me take one fungus at a time and then we'll talk about it. But please don't forget to remember and revise this one table. Is that clear? This one table you should be revising a day before. Clear? Okay. Now coming on to the first class. Yes, my question to all of you is, what is the basis of classification of this fungi? It is a very important previous year question. Yes, anybody who remembers? What is the basis of classification of fungi? Very important, basis of classification of fungi. Yes, please. Number 1, number 2, number 3. Type of spores. Yes, types of spores, or you can say fruiting bodies. Yes or no? So fruiting bodies are the structures inside which spores are formed, right? Then type of mycelium. Very right, type of mycelium. And what is the third one? Yes, one. What is the third basis of classification? All of you, quickly let me know. Reproduction, nutrition, mode of spore formation. See, type of spores, or you can write the fruiting body separately. Fruiting body, because spores will be formed inside these fruiting bodies only. So type of spores, type of fruiting bodies, and the type of mycelium, these are the three bases of classification. Are these points clear to everyone? Please quickly let me know. Let me see your chart now. I hope nobody is wasting their time and everybody is writing these quick points along with me. Quickly let me know. Give me a thumbs up that we are all full of energy and everybody is writing the points with me. Quickly. Yes, please. Let me know. Let me know. All going well? Perfect. 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 Yes. Green hearts. Wow. Okay, Nandu. All right. Okay, let's continue. Let's continue. So see, what are the points that you must remember? So Phycomycetes is one fungus which is aseptate and cenocytic. You will find them in aquatic, damp areas and decaying soil. Asexual spores, I told you, motile. If motile, they will be zoospores. If non-motile, aplanospores, and sexual ones are known as zygospores. Examples also I'll tell you how to remember. There is second very important table that we are going to discuss today. Ascomycetes. So Phycomycetes, by the way, are also known as algal fungi. They are also known as algal. Phyco word is for algae. Algal fungi. Clear? Ascomycetes are also known as sac fungi. And next point, okay. They are septate hyphae. Asexual reproduction by conidia, sexual spores by ascospores, formed inside the ascus and ascocarps. Now, what is the difference? See, I'll tell you. So this is the hypha, and the hypha is septate. I'm talking about Ascomycetes. This is a septate hypha. So what happens for asexual reproduction? One of the hypha grows vertically upwards and the tip swells up. Okay, and then it divides and divides and divides. So these are conidia. These are conidia. So do you see that conidia formed on the outer side? This is an asexual spore. While in case of, let's talk about Phycomycetes, they are aseptate, right? They are aseptate. So aseptate are always cenocytic. Cenocytic means multi-nucleated condition. So this is an aseptate hypha. So one of the hypha grows vertically upwards. It swells, right? This swelling is known as sporangia. This swelling is known as sporangia, and inside these sporangia are formed the spores. So spores are not produced outside. Spores are produced within this fruiting body, within this structure known as sporangia. So that is why we call these spores. These spores can be zoospores also, or they can be eplanospores also. If motile, then zoo. If non-motile, then eplanospores. But they are both produced within this sac-like structure called sporangia, and that is why we said these are endogenously produced. While if we talk about conidia, conidia are produced on the outer side, they are exogenously produced. Now, is it clear? All those of you who had this doubt, what do we understand by exogenous or endogenous? Are these points clear? Okay, coming on to Basidiomycetes. Now, Basidiomycetes are also known as club fungi, right? They have basidium, and inside the basidium are formed basidiospores, and basidiocarp is the fruiting body. Are these points clear? And they don't have asexual spores generally. And then Deuteromycetes, we call Deuteromycetes as fungi imperfecti because they do not show. Why are they imperfect? Because of no sexual stages. No sexual stages. Clear to everyone? And then only asexual stages are known to them. By the way, later on it was discovered that there are some sexual stages present, but they don't really resemble the characteristics of Deuteromycetes. So the sexual stages were they were placed separately under, placed under Ascomycetes or Basidiomycetes. Another question. They were placed under Ascomycetes or Basidiomycetes. Are these points clear? Very important question. That the sexual stages of Deuteromycetes are placed under which two classes? Asco and Basidio. Is this point clear? Everything will be covered. Just wait. My way is different. I'll be taking different things at different times. I will be discussing all the diagrams at the end. So don't worry about anything. Is that clear? Don't worry about anything. So next point that you must know, how to remember the examples. So we have pad B, right? So how to remember the examples? So for Phycomycetes, we must remember pram. We must remember pram. For Deuteromycetes, you have to remember D-cat. And for Basidiomycetes, you must remember Bapu. Bapu is father, right? Okay. So for pram, we have, now see, these are the mnemonics. Okay. So for pram, we have Rhizopus. We have Albugo, which is parasitic over rye, over mustard, right? And then Mucor. Are these all points clear to everyone? So these are the examples. So how do we remember the examples of Phycomycetes? Pram, Rhizopus, Albugo, Mucor. Then for Deuteromycetes, it is DCAT, Coleotrichum. We have Alternaria. Then we have Trichoderma. Phyco में क्यों नहीं रखा? Because the characteristics didn't match Phycomycetes. The characteristics of sexual stages were more like Asco and Basidio. Then for Basidiomycetes, we have Bapu. So we have here Agaricus, your mushroom, or toadstools also come under this category. Agaricus, then Puccinia, and then Eustilago. Is this point clear to all? Now, which of them are parasitic? Remember, these are parasitic over rye. Parasitic over rye. Rye is mustard. Clear? Now, Alternaria, these are all parasitic. Then Puccinia and Eustilago, they are also parasitic. How do you remember? What do they cause? So, for remembering Puccinia, we have another mnemonic, Puccinia and Eustilago. smut, and this causes rust. So how do we remember this? USPR. Everybody wants US, United States permanent residentialship, right? So USPR. So Eustago causes smut and Puccinia causes rust. Are these points clear to everyone? I don't remember. I don't understand Telugu. I can teach on smartboard. I have smartboard, Chandresh, but as I already explained, it will take me another 15 days and we'll be back on the board. But till then, let's adjust. Let nothing stop our revision. Okay? All right. Coming on to, okay, we are done with all this. Now the third, sorry, Ascomycetes. So the examples in Ascomycetes, all the other examples in fungi which are left in NCERT, they are all under Ascomycetes. What are those? Ascomycetes have Neurospora. So now the economic importance of these Ascomycetes is very important. Neurospora. What is this used for? Biogenetic studies, biochemical studies, biochemical genetic studies. Okay, then we have Aspergillus. Aspergillus produces citric acid, if you remember, Aspergillus niger, right? Citric acid, source of citric acid. Yes or no? Then we have penicillium, which is the source of penicillin, an antibiotic. Then it has Saccharomyces, the unicellular fungi, Saccharomyces, the baker's yeast or the brewer's yeast, remember? I'll keep asking you whether you remember or not because it is all about what you remember. So that is why I have brought these point-wise notes so that you are able to revise, recollect, okay? Then penicillin, Saccharomyces, then we have claviceps. Now claviceps is that fungus also known as ergot fungus which is the source of LSD. Do you remember the most potent hallucinogen? Yes or no? Any other example? Yes, Neurospora, Aspergillus, Penicillium, Saccharomyces, Claviceps, and very, very important, morels and truffles. Morels and truffles. These are two such Ascomycetes which are used as delicacies. These are used as delicacies. Yes or no? All clear? Perfect. Now, moving on to the next structure, which is virus. So let's talk about virus. So virus, see here. Okay. Now virus is acellular. Virus is acellular. Why? Because this body is not really made up of cell. It is a connecting link between living and non-living. Yes or no? Connecting link between living and non-living. Inside the living, it will behave as living. Outside the living host, it will behave as non-living. Can anyone tell me which is the living character of virus? Yes, please. Which is the living character of virus? Yes, please. Living character of virus is reproduction, division. Yes or no? Living feature of virus is reproduction. Now, it will be living only inside the living host. So, if it has to prove itself to be living, it absolutely needs a living host. So, they are obligate parasites. This is another feature that you must know about viruses. They are obligate parasites. There is no other way in which they can survive. Is that clear? Okay, then they will either have DNA or they will have RNA. No virus will have both DNA or RNA. This is NCERT line. Yes or no? Okay, then see, virus is made up of two components. There are two components around the virus. This is the protein covering known as capsid, and inside it has the genetic material. These are the only two things which a virus consists of. Right? Capsid, the cover, which is made up of protein, and genetic material will either be DNA or be RNA. There is no such virus till now which has both the genetic material. Now, this capsid is made up of smaller units, and the smaller units are known as capsomeres. Are these points clear? Clear? Okay. Moving on to the next, we have, so capsid I've talked about. We've talked about capsomeres. Now these capsomeres, these proteins are crystalline. So what is the non-living feature? What is the non-living feature of virus? That it is crystalline in nature. It forms crystals, crystallizable. So you must know which feature of virus is living and which is non-living. Are these points clear to everyone? And they are all obligate parasites, so they will obviously cause diseases. They are all pathogenic. Can anyone name the the diseases caused by virus? Yes, please. Anyone? Special classes timings will be informed very soon. See, this is my first class. Let things get into flow. In 1 or 2 days, we'll start with the special classes also. Probably from tomorrow itself. I'll inform you the time. Okay, common cold, dengue, chikungunya, flavivirus, right? Rhinitis, then HIV/AIDS, herpes. These are all viral diseases. Clear? Yes. Talking about TMV. Now, TMV was the first virus to be discovered which has RNA as a genetic material and the capsid is in the helical form. Okay. Now, talking about the three scientists. Remember, when we are talking about viruses, we must remember three scientists. One is Dmitry Ivansky. Second was Bejernik. And the third one was Stanley. Now, what did Ivansky did? Discovered virus. Which virus? TMV. Okay. Bejernik named the term, or he coined the term virus. Bejernik coined the term virus. He also called virus as contagium vivum fluidum. Are these points clear? Contagium vivum fluidum. So he called virus as venom, poisonous infectious fluid. Is that clear? Poisonous infectious fluid. And then Stanley was the first person to crystallize the virus. Are these all points clear to everyone? Are these points clear to everyone? Please give me a thumbs up that you're making these notes, quick notes, side by side. Everybody is here with me? Yes, please. Everybody is here with me? Now, special virus. Yes, special viruses which will attack. Special viruses. See, there are viruses which are going to attack animals. Now, another thing, viruses that you must remember, the viruses which attack animals are known as animal virus. Then we have plant virus. And we have bacteria infecting virus, which is known as bacteriophage. Okay, everybody take this homework. Very important. Now, see, take it as a homework. Find it out, all of you. I'm going to ask this in the next class. This is your homework. You have to find out the genetic material present in each of these viruses. You have to find out the genetic material in each of these viruses. Yes or no? How many of you are going to do this? How many of you are going to do this? The types of genetic material. See, I'm going to talk about bacteriophage here. Bacteriophage infects the virus. It has double-stranded DNA. One homework is done. It has different parts: head, tail, sheath, and tail fibers. These are the 4 parts of bacteriophage. Is that clear? And then, other than virus, we have viroids. Now, what is a viroid? Viroid is only RNA. It is free RNA. There is no protein coat. Is that clear? So the molecular weight is obviously low. It has molecular weight which is lower than the molecular weight of virus. Smaller in size, and it is a plant pathogen. One very important example you must remember, it causes disease, tobacco. spindle tuber disease. Tobacco spindle tuber disease. This disease is caused by viroid. Then similarly, we have prions also. Now in prions, there are only proteins and we don't have the nucleic acid. In viroid, there is nucleic acid but no protein coat. In prions, prions, P for proteins. It has a protein coat. Is that clear? And it causes neurological diseases like mad cow disease. Mad cow disease. Okay, now it is also known as Creutzfeldt-Jacobs disease. Is that clear? Mad cow disease is bovine, means it affects cows, while Creutzfeldt-Jacob disease is a variant which causes it in humans, the disease that causes it in humans. Now the virus is bovine, the name of the prion is bovine spongiform encephalopathy. It causes this disease, encephalopathy. Are these points clear to everyone? This bovine spongiform encephalopathy is known as mad cow disease and its variant causes a similar disease in humans known as Creutzfeldt-Jacobs disease. Are these points clear? So now let's quickly revise the two kingdom classification which was given by Carolus Linnaeus, right? And the basis of classification was presence or absence of cell wall, mode of nutrition, autotrophic or heterotrophic, and motility. So he gave two kingdoms as Plantae and Animalia. Is that clear? The limitations, the disadvantages of this was that there was no differentiation between prokaryotes and eukaryotes. This was a criticism of two kingdom classification. Criticism of two kingdom classification. Right? No differentiation between prokaryotes and eukaryotes, unicellular or multicellular, photosynthetic and non-photosynthetic. Clear? Then fungi was also placed along with the plants. Are these points clear? Cyanobacteria was also placed along with the plants. Heterotrophic bacteria was also placed along the plants. Are these points clear to everyone? Then we got three kingdom classification. Three kingdom classification was given by Ernest Haeckel. And the third kingdom which was actually added in this was Protista. Now what did Haeckel do actually? Haeckel separated the unicellular organisms. He took out unicellular organisms from Plantae and Animalia and made a separate kingdom called Protista. See, separation of unicellular organisms. Okay. Now limitation, still another criticism that he could not separate prokaryotes and eukaryotes. They were still put together. Then we got four kingdom classification by Copeland. And the fourth kingdom which was added by Copeland was Monera. So what did Copeland actually do? He separated the prokaryotes. He said, okay, if Haeckel could not separate, I am going to separate prokaryotes from eukaryotes, and he made a separate kingdom. Are these points clear to everyone? Are these points clear to everyone? Okay, perfect. Then we got the five kingdom classification, because still in all these three or four kingdom classifications, fungus was still not separated. So R.H. Whitaker actually made a separate separate kingdom for fungi. So Whitaker introduced fungi. Is that clear? Please understand this. What are we studying here? Who introduced? Okay. Okay. So Ernest Haeckel introduced Protista, Copeland introduced Monera, and Whitaker introduced fungi. Are these points clear to everyone? Okay. Now, what are the bases that I have already discussed with you? These were the bases. After five kingdom classification, we got six kingdom classification. Six kingdom classification was given by Carl Weiss. Now, what did Carl Weiss did? Carl Weiss actually divided Monera. He divided Monera into two, Archaebacteria and Eubacteria. And so overall there were six kingdoms. One was Archaebacteria, Eubacteria, Protista, fungi, and so on. Now these six kingdoms were divided into three domains. So six kingdoms were divided into three domains. Archaebacteria was the first domain. Eubacteria made the second domain. And the rest of them, all the eukaryotes, were put together in the third domain. And we called them eukaryota. So first and second was prokaryota. Ishwar, can you stop spamming, please? This was very irritating. Don't spam. Please don't spam. Or you will be chucked out of the class. I'm so sorry, but this is the norm. Nobody is allowed to spam in the class. Please be very serious. Okay? Yes, please. So, how many domains were how many domains were given to the prokaryotes? Two domains: archaebacteria, eubacteria. And how many domains were dedicated to eukaryotes? Only one domain. Are these points clear to everyone? Perfect. So with this, guys, we complete our first chapter, that is biological classification. Now coming on to the next, which is Porifera, animal kingdom. And we are going to start with the first phylum, which is Porifera, commonly known as sponges. Commonly known as sponges. Is there any doubt? You will get all these slides, you will get all these PDFs. Is there any doubt in what we have read so far? Please let me know quickly. Any doubt? Or give me a thumbs up that you're all clear with what we have read till now. How helpful is the session is to revise? How helpful has the session been to revise everything from the previous chapter? Please let me know. No doubts? There's one point called lichens in the previous chapter. See, lichens we keep on talking about in other chapters also. So lichens is basically a symbiotic association. It is a symbiotic association between algae and fungi. Algae and fungi. Now we call algal partner as phycobiont. See, let's quickly revise this. Phycobiont and fungal partner as mycobiont. Now, since it is a symbiotic association, both are interdepending on each other, but still mycobiont is known as the dominant partner. This is still known as a dominant partner out of the two. Okay. Now, phycobiont, it is an algae, so it has pigment, it can photosynthesize. It is going to be photosynthetic. While fungi, it has a thalloid body and it is going to bring carbon dioxide. It absorbs carbon dioxide and water. So, fungi absorb CO2 and water while algae photosynthesize the food, and this is how they live happily ever after. Are these points clear? Perfect. Also, what we must know about lichens is that lichens are the hashtag bioindicators. They are the bioindicators of SO2 pollution. So, if you ever see that white deposition on the tree bark, you should be happy that you're living in a sulfur dioxide free area. Is it clear? It comes under mutual. See, mutualism is of two types. Mutualism is of two types. One is symbiosis, second is proto-cooperation. It comes under symbiosis. Every symbiosis is mutualism, but every mutualism is not symbiosis. So yes, they are the bioindicators of SO2 pollution. Are these points clear, everyone? Perfect. Now let's move on to the next topic, that is Porifera from animal kingdom. So we are going to quickly revise. They are mostly marine, but they are not always marine. So I'm giving you another homework. See, I am going to give you homework. I'm going to ask you that also. And I'm going to give you the answers also in the coming session. Another homework for you is you have to list down. See, you have to list down all the organisms which are exclusively marine. Which are the phylum which are exclusively marine? This is going to be your second homework. Very easy. Okay. Now, they are mostly marine, but some are freshwater. For example, Spongilla. That is why Spongilla is known as freshwater sponge. Do you remember that? This is also known as the freshwater sponge. Clear? Now, this is the only phylum in the entire animal kingdom which has cellular level of organization. The body is usually asymmetrical, means it cannot be divided into n equal halves along any plane. Then body has many pores. So this is a unique feature. You will not see this happening anywhere else. Body is made up of so many pores, and the pores are known as ostia. And these pores lead to the water inside. So another unique feature is the water canal system. So in the water canal system, what is the route of water? You must know that. The route of the water is water enters through ostia, goes into the incurrent canal, then into the spongocele, and from spongocele it goes out into the osculum and then outside, exit. Are these points clear to everyone? Are these points clear to everyone? Sure. Moving on to the next. So it has many pores. It has water canal system. It has water canal system. Clear? Then the water exits through osculum. Already told you water will exit through osculum. The central cavity is known as spongocele. And this spongocele is also known as paragastric cavity. It is also known as paragastric cavity. So if I draw this, so it is like, so this is the body of a sponge. This is the body of a sponge, right? And see, so there are two layers. These are the pores. This is the incurrent canal, right? This is the spongocele. This is the osculum, and the water goes out. Are these points clear to everyone? Paragastric cavity, perfect. Now, these have very unique cells known as conocytes or collar cells. Now, where are these collar cells present? These collar cells are present all along the spongocele. These are the collar cells. Okay. Now, collar cells are flagellated. These are flagellated. So basically, they are going to circulate the water within this cavity. And this circulation of water, just like it happens in the washing machine, right? So they're going to circulate water in this cavity, and this water will bring food. This water will bring sperms. It will carry out the eggs. Are you getting it? It will take out the waste matter. So this circulation water is important, and who circulates it? The flagella of the collar cells. So it lines the entire spongocele. This is an important line of NCERT. Conocytes line the spongocele. They help in the water flow. Digestion is of intracellular types. Now, very important, the skeleton is made up of spicules. Now, where are these spicules present? See here. So, this space between the two layers is known as mesohyl layer, right? This is known as a mesohyl layer. And here we have these skeletal structures known as spicules. These are spicules. Is that clear? Now, these spicules can be made up of calcium carbonate or they can be made up of silica. These spicules can be made up of calcium carbonate or silica. Now, the second, see, spicules is first skeletal structure. The second skeletal structure is spongin fibers. The second skeletal structure is spongin fibers. So, there are two types of skeletal structures in Porifera. Is that clear? Yes, please. Anyone, where can I get these notes? You will get the notes in the description box. Let me complete the class. I'll attach the PDF in the description box. Okay? All right. Then, but the best notes are the notes which you are making yourself. Of course, you will get these notes, but you should be writing them down yourself. Now, there are no tissues. That is why we say it has cellular level of organization. Now, the exchange of gases or waste occurs by diffusion through body surface. Through body surface. Are these points clear? Yes or no? Now, this is the homework. Now, it shows asexual reproduction by budding, fragmentation, or gemule formation. Now, see, gemules are special asexual buds. Asexual, and these are internal buds. And they are only formed in Porifera. You will not find it out in any other phylum of animal kingdom. Sexual dimorphism, yes. Sexual reproduction, yes, it occurs. It is a hermaphrodite, so it is a bisexual organism, right? And then indirect development occurs and the larval stages are present. Are these points clear? And the fertilization is internal fertilization. Now, all those of you, I'll promise you one thing. I'll promise you one thing. Listen, listen, listen. All those of you who will attend this whole lecture with me and they are writing down the points with me, you will get a complete flow chart, detailed flow chart of animal kingdom from my side in the special class. So learn this chapter, revise this chapter, attend my class. We will discuss so many questions in that class and statement based questions, the new type of questions which NTA is giving these days. Okay. And one bonus thing would be I'll tabulate everything in a animal kingdom and I will share it with you in my special classes. Okay. So how many of you are going to learn these chapters and revise and come to my question based special classes? Yes, please. Quickly let me know. Where to attend, Jyoti? It is on the Unacademy app. Okay, all of you are going to do that. Yes. And one more thing that I wanted to share with all of you, very, very important. See, it is super, super important that, see, last few days are left. All of you must take these mock tests daily. Everybody, starting from 19th of April, you have to take these mock tests daily without any gap. You don't have to pay a single penny. So, okay, you don't have to pay, but they're free of cost. There will be real neat exam experience. You will get your rank also, instant AIR, all India rank will be predicted, so that you know where you stand, where your preparation stands, where you are lagging behind. You will compete with lakhs of students. Video solutions will be available after every test, and top 50 students will get mentorship after every test, personal mentorship, right? And top 10 students will join the WhatsApp guidance group where you can put down your doubts. And the top 3, see, top 50 will get mentorship, top 10 WhatsApp guidance, and top 3 will get 500 worth Amazon voucher. Now these are absolutely free and they are starting from 19th of April. Timing will be exactly the same, which is the real NEET exam timing. Okay, so that you actually get the real NEET exam experience. Are these points clear to all? So how many of you are going to do this? How many of you are going to do this? Quickly let me know. See, if you will not take these mock tests, sorry to say, but sadly, it will reflect in your preparation. Rather, I feel, I remember my childhood, to be honest. My mother would actually make those papers, and be I would have to write down everything. I would have to take a paper before my real paper. And that has always helped me. It has always given me confidence. You also know what is the importance of mock tests. So take this mock test on all India level. Get your rank. See where you really stand. Okay? Perfect. Now, start talking about the examples, Cycon, Spongula, and Euspongia. Now, Euspongia is known as the bath sponge. This is known as bath sponge because it is used for bathing purpose. Why? Because it don't have spicules. Now, spicules are hard. Spicules are absent. It only has spongin fibers which are softer, right? So, it can be used for bathing purpose. Spongin fibers are soft. They can be used for bathing purpose. Rest, all of them, they have spicules and spongin fibers. Are these points clear? Perfect. Then coming on to Cnidaria or Coelenterata. The unique features, now see, not the unique, the general features first. They are aquatic, they are marine, some of them are freshwater. Okay, for example, Hydra. They are diploblastic. Porifera is also diploblastic. There are two germ layers, ectoderm and endoderm. Then, now this shows the tissue level of organization. Now it has tissue level. Porifera has cellular level. See, there are some words, as soon as you hear those words now, things should just click. For example, let's revise. Soap box. Where did you read about soap box? In the entire biology, where is soap box thing coming from? Quickly let me know. When you think of soap box, when you hear soap box, what should click? If you can answer these questions instantly, only then your paper will be clear. Yes. See, I am a bubble burster. I'm known for bursting bubbles. If you know me in my personal life, I'm a bubble burster. I don't elevate you. I don't keep you just floated. Okay, ho jayega. You will be able to. No, until and unless you change your habits, until and unless you make those efforts, you put in those efforts, things will not happen in your favor, right? So yes, These things have to click. That is why I'm bringing these one word like notes. Okay? Yes, soap box thing. You read about soap box like structure only and only in diatoms. Perfect. Similarly, cellular level of organization, you will only read about it in Porifera. Is that clear? Cellular level of organization, mules only in Porifera. Clear? Perfect. Then radial symmetry. Now, what do you understand by radial symmetry? It can be divided into equal halves along any plane. Any plane. Okay, the body wall, we can skip this body wall. Now, coming on to, see, there is gastrodermis, you can leave it. Now, the name coelenterata comes from gastrocoelentron. Now, coelentron is a gastrovascular cavity. That means it will function as an alimentary canal also and the circulatory system also. Are you getting that? It will function as a transport system, circulatory system, as well as the alimentary canal. So one cavity which will serve both the purposes, gastrovascular, and we call it coelentron. Coel means cavity, entron means alimentary canal. Is this clear? But it has a single opening which will serve as both mouth and anus. So that is why it has incomplete alimentary canal. See, Porifera doesn't have alimentary canal at all, while this one has, and it has incomplete alimentary canal. Single opening for ingestion as well as for removal. Then there are special cells called cnidocytes or cnidoblasts. They are present on tentacles and all over the body. Okay, and all over the body. Now these cnidoblasts perform defense, capture of the prey, and they also help in anchorage. There are three functions which NCERT has mentioned about cnidoblasts. Stinging cells. These are the stinging cells. They sting. These are the stinging cells. They sting. They actually sting. So they are used for defense. They are used for capturing the prey. Now digestion here is both extracellular and intracellular. While in Porifera it was only of intracellular type. In Porifera it is only of the intracellular type. Now the coelenterates are not very boring. They have a very interesting lifestyle. What is so interesting? They have two body forms. See, we are born and we'll die in this body form only. But when we talk about coelentrates, they have two body forms. Not only two, it can be more than two. 2 to 5 body forms have been seen in coelentrates. So they have multiple avatars, right? So this phenomena is known as polymorphism. This is known as polymorphism. This is only and only seen in coelenterate. You hear about polymorphism, you should think about coelenterata. And what are the two body forms? Polyp and medusa. Polyp and medusa. Now this polyp and medusa, let me write down here only. These polyp and medusa, we call polyp as the asexual form. Why? Because it reproduces asexually. Medusa is the sexual form because it reproduces sexually. It will form gametes. Second thing, polyp is plant-like. It is sessile. What is plants? They are attached to one surface. They don't move. They are sessile. Now, being a plant-like, it is nutritive in its function. It provides nutrition. While Medusa is free swimming. If you have seen jellyfishes in TV, right? Jellyfishes are swimming, right? Free swimming. Jellyfishes that represent Medusa state. They are free swimming. They are defensive. They provide defense. Is that clear? They are motile. So this is all about Medusa. Now, one form can change into the other. This Medusa can change into polyp. Polyp can change into Medusa. Now, this thing is known as metagenesis. So, the sexual and asexual forms will alter with each other. This is called metagenesis. Are these all points clear? Right? Then reproduction will be asexual through budding and sexual also. And these organisms are also bisexual. Sorry, they are also bisexual, means hermaphrodite. Sexes are not separate, they're present in the same body. Then they also show indirect development, and fertilization is external. Is that clear? No circulatory and excretory organs. Now, what are the examples? We have freshwater hydra, Aurelia, We have Obelia, known as the sea fur. Physalia, the Portuguese man of war. Very important example, Portuguese man of war. Adamsia is the so this is a jellyfish. Aurelia is the jellyfish. Adamsia is the sea anemone. Pennatula, the name itself says, it looks like sea pen, right? Gorgonia, and then we have Meandrina is the brain coral. Yes, please. Let me know. Gorgonia is sea fan. Yes, this is sea fan. You must remember the common names along with the scientific names. Both the names should be clear to you. Are these points clear? Now, coming out of Coelenterata was another phylum, Ctenophora. We call them comb jellies. We also call them sea walnuts because they look like walnuts. We also call them sea gooseberries. Gooseberries are amla. So they look like amla. So sea gooseberries as well. Is that clear? Now, exclusively marine. See, I've given you this homework. You can actually keep doing that homework side by side. Write it on the top of your page. Write it on the top of your page. Exclusively marine. This is your homework. The first exclusively marine is comb jellies. Radially symmetrical, diploblastic. Now the unique feature here is that they have 8 rows of comb plates, ciliated comb plates, which are meant for locomotion. They have coloblasts. Sorry. They have coloblasts. Coloblasts are the adhesive cells present on the flagella which will pick food for capturing the prey. Now digestion again like Coelenterata is extracellular and intracellular. Clear? Yeah. Now another unique feature, they are bioluminescent. What was another structure that we talked about was bioluminescent? Yes, anybody? Let's keep revising along with another bioluminescent organism that we have talked about in this session. Yes, please. Emit light, yes. But which another organism have we talked about? Shrivan, this is not time for extra out of books. This is the time to revise what is given in NCERT and recollect that. You never need extra. You need this thing properly inscribed in your memory. Yes, dinoflagellates, goniolids, remember? Red tides. So all those, they were bioluminescent. And this is also hermaphrodite, means bisexual development. Here also development would be indirect. Sexes would be united. That is what in hermaphrodite is. Then fertilization here also would be external, right? And the examples are pleurobrachia and tenoplena. Coming on to platyhelminths, also known as flatworms because they have a dorso-ventrally flattened body. That is how they get their name. Now, these will have bilateral symmetry. They are the first triploblast. The first one to have organ level of organization. They are acoelomates. Now, all the acoelomates together, yes please, all the acoelomates which do not have coelom. So, Porifera is an acoelomate. Coelenterata, remember? Ctenophora, these are all acoelomates. And now, platyhelminth is also a acoelomate. Anyone with any doubt? These are acoelomates, they do not have coelom. Now, the first three are diploblastic, and this one, platyhelminths, being a triploblastic, the first triploblastic, but still doesn't have a coelom. Are these points clear? Now, mostly endoparasites, so they live inside the living host, and hooks and suckers are the adhesive structures. So they help, hooks and suckers help the parasites to attach to the body, adhesive structures. Are these points clear to all? How we listen special class? You don't have to listen, you have to attend the special class, and you have to solve important questions with me. And some of the questions which I will be solving in the special class, some of them I have brought here also. So let's quickly complete the chapters and we will be jumping on to that as well. Okay. Now, digestive system is incomplete, so there is no anus, single opening which will function as both mouth and anus. Flame cells, another unique feature. These flame cells are only found in platyhelminthes. So the moment you think about, you hear flame cells, the fire that that should burn inside you is okay. We are talking about platyhelminthes. Are you getting it? So flame cell is also one such word which quickly brings the memory of platyhelminthes. Are these points clear? Then these are meant for osmoregulation and excretion. Hermaphrodite, so it is a bisexual, mostly internal fertilization. Okay. And then remember, till now the body of all these phylum have been unsegmented. Okay. So unsegmented bodies. Unsegmented bodies. So will be found in which all phyla? Yes, unsegmented bodies. We'll have Porifera, I'm writing in short. We have Coelenterata, unsegmented. We have Ctenophora, unsegmented. Then platyhelminthes are also mostly unsegmented. Tapeworm is an exception which shows false segmentation, pseudo metamerism. Otherwise, mostly platyhelminths are unsegmented. Aschelminthes, absolutely unsegmented. Okay, then mollusca, sorry, not mollusca. Mm, 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 mm. Okay. Are these points clear? Yes, monaceous is hermaphrodite only, Vishwa. Monaceous is a term used for hermaphrodite only. Monaceous, bisexual. Clear? Now, planaria is also coming on to that. So this shows that your revision is going really well. Now, life cycles are often complex with the larval stages. So this is a parasitic feature that they have complex life cycle. Life cycle means many larval stages. Many larval stages. Are these points clear? Points clear? Okay. No, Shri, you are wrong. Shorter male and female longer. This is an estelminthic feature. Okay. Anyways, coming on to platyhelminthes, Taenia, fasciola, planaria. Planaria is also known as Deutesia. Well marked regeneration. Well marked regeneration. Is this clear? While these both are parasitic in nature, they cause diseases. They cause diseases. Tapeworm will cause, or Taenia will cause teniasis. Fasciola will cause fasciolosis. Hanji. Fasciola will cause, yes, liver rot. How many marks can we get if you follow our notes? Siva, you are going to revise everything. Every point in NCERT will be revised in these lectures. So if you revise everything and you're able to recollect everything in the exam, 360 out of 360 is the answer, right? 360 out of 360. So see, these last 20 days are very, very crucial. If you will revise everything, you will get 360 out of 360. Have you heard about adrenaline rush? Let me tell you one more function of adrenaline. Nowadays you are in that adrenaline rush. Make good use of that. Adrenaline causes short term memory also. See, there are certain things that are already well learned by you, but then there are certain things that you keep on forgetting. But this adrenaline is going to help you remember those things also. So make good use of this time. So this adrenaline kick is going to kick off your preparation also. Okay, now coming on to Aschelminthes, also known as nematoda or nematihelminthes or roundworms. Why do we call them why do we call them roundworms? Because the body is rounded in cross section. It has a isodiametrically round body. Isodiametrically round body, as if somebody has carved it with the help of a compass. It is so round. That is why the name is roundworm or nematode, also known as threadworms. Body is very thin. Now they are triploblastic, they are bilateral symmetry, and they are pseudocoelomate. So after platyhelminthes, which is an acoelomate, the first coelom that will appear is a pseudocoelom. Again, pseudocoelom is such a word. The moment it comes in front of you, you should only and only think about Esculmenthes. Are you getting that? So what is pseudocoelom? So in pseudocoelom, basically, mesoderm is found as scattered pouches in the coelom. Remember, what is pseudocoelom? Mesoderm as scattered pouches. Pushpa, you don't have to be in it. I am just telling the students, in case you are in adrenaline rush, then it will help you. If you are not and you're very calm and composed, which is a beautiful thing, but it is rare that exam is right around the corner and you are very calm and composed. In case you are that one personality who can stay composed during the exam, then trust me, you will not forget what you have learned. You have already learned things. It will go in a long term memory. The only difference is going to be short term and long term, but that short term will extend till 3rd May. Don't worry. Then body round and tapering at the ends, body covered with cuticle. This cuticle is a parasitic feature and prevents it from getting digested by our enzymes. Then coming on to organ system level of organization, first appeared here. Then complete digestive system first time appeared in Esculmenthes. In Platyhelminthes, it will be either absent or it will be incomplete. In sexes will be separate. And for the first time, sexes are getting separated, means they are unisexual, they are dioecious, male, female separate, and they can be distinguished on the basis of external features. They show sexual dimorphism. Fertilization is internal, mostly parasitic, some are free living, right? Ascaris, the common round worm. Then we have Wuchereria, the filarial worm, which causes elephantiasis. Remember? Can anyone tell me how is Wuchereria going to enter your body? Ankylostoma, the hookworm. Entrobius, the pinworm. You can skip this one. Entrobius. How does filarial worm enter your body? By the help of a mosquito known as Culex, female Culex mosquito. Female Culex mosquito is the vector. It will bring Wuchereria in our body. It will lay in our lymph vessels and it will block the lymph vessels, causing the disease called elephantiasis. Yes? Or filariasis. Yes or no? Perfect. You remember these things. See, why am I asking these questions from different chapters? Because this is what is needed. You need to connect dots. Is that clear? All right. Come moving on to the next phylum, which is Annelida, or we call them segmented worms. First time you will see true segmentation here. They will be triploblastic, bilateral symmetry, true coelom. Then metameric segmentation is a true segmentation. True segmentation means the number of segments in the body will remain fixed from birth to death. Number one. External and internal segmentation will coincide. That's what metameric segmentation is. Is that clear? Okay. Moving on to then more, again, it also has organ system level of organization, body covered with thin cuticle and, okay, wait, thin cuticle. Okay, guys, there's a danger in this room. There's a wasp. Anyways, it has closed circulatory system. The first time closed circulatory system is going to appear in Annelida. Before this, now see, before this, Porifera, listen. Porifera, Coelenterata, Ctenophora, Platyhelminths, Esculmenthes, then we'll have Annelida, Mollusca, Arthropoda, first of all, Arthropoda, then Mollusca, then Echino, then Hemi, and then Chordata, right? Let me give you the general features here. About the circulatory system is absent here. Circulatory system is absent here. First time it appeared in Annelida and it had closed circulatory system. Is this clear? Then Arthropoda had open, Mollusca usually open, few have closed, Echino open, Hemi open, in Chordata mostly closed. Is this clear? Mostly closed circulatory system. Now, all those of you who have been attending the class since the beginning must know that I have told you to attend these mock tests. See, now the pointers that I'm giving you, whether you remember or whether you are able to, you know, retain them or not, how will you get to know? How will you be able to practice connecting the dots like I'm asking you questions in the session from these all India mock tests? So make sure that you don't miss even a single mock test starting from 19th of April, every day 2 to 5 p.m. No classes will be kept within this time period so that all of you can attend. These are free mock test series. Everybody has to take it. So this is a break only, Shri. This is a break only. When I'm giving you gyan, how to attend the paper, things you should take care of. That is a break, isn't it? We can't afford any other break. We don't afford any other break. And there's a wasp right in front of me, dancing and trying to tell me that it can bite me anytime. But am I scared? Little bit. Okay. Are these points clear? Continue till 7:10? No, no, I'll continue beyond that. See, there are 4 chapters. We'll need time, isn't it? Now, see, coming on to, yes, so Annelida has a closed circulatory system. Respiration will be through skin or gills. Wait a second. The respiration will be through skin or gills, particularly mostly skin, which is known as cutaneous. Mostly known as cutaneous. Are these points clear to everyone? Okay. Now, excretion is through nephridia. Locomotion is through septa or parapodia. Nervous system, it has ventral nerve cord, which is which is ventral, which is solid, and which is double. There is some lag. Wait. See, for enrolling in the mock test, Sashmita, the link is given in the description box. So you don't have to hunt far. The description, it is all given in the description box. Okay. The nerve cord is ventral, it is double, and it is hollow. Clear? Reproduction is sexual or it can be asexual also. Examples are Nereis, Peritima, and Hirudinaria. Now, about Nereis, see, we need to remember certain things. About Nereis, you need to know that it has some special structures called parapodia. Nereis has parapodia. These are paddles which it uses for swimming. So it is aquatic. While these two are terrestrial. These two are terrestrial. Are these points clear to everyone? Earthworm, you know, is the friends of the farmer. Leech is blood sucking. So we call this blood sucking as sanguivorous. It is blood sucking. It stays outside the body and sucks blood. So it is not an endoparasite. It's an ectoparasite. Next unit will be morphology, anatomy, and structural organization. Thank you, Shri. Thank you. So don't forget to share these sessions with all your friends as well. Okay. Now, coming on to the next phylum, which is Arthropoda. Largest phylum, triploblastic, bilateral symmetry, segmented body, and the body has 3 segments: head, thorax, and abdomen. Head, thorax, and abdomen. But sometimes this head and thorax can be combined to form cephalothorax as well. Are these points clear? See, jointed appendages, chitinous exoskeleton, these are the most important features of Arthropoda. Are these clear? Open circulatory system. Now, open circulatory system means there are no capillaries. So, wherever there is empty space, blood will flow in. So, blood fills the whole space. See, blood fills the whole space, fills the coelom. Now that is why such coelom is known as hemocoel. The coelom filled with blood is known as hemocoel. Such cavity will be known as hemocoel. Respiration can occur by three methods. It can have gills, book gills, book lungs, and tracheal system. Are these points clear? It has, it has book gills, book lungs, and tracheal system. Just let's cut this out. Okay. So book gills, book lungs, and tracheal system, these are three methods of respiration. Excretion can occur through through malpighian tubules, coxal glands, and green glands. So different arthropods will have different methods for respiration and excretion. So it has compound eyes, sexes are separate. If you remember cockroach, male females were different, right? Development will be often indirect but can be direct also. Fertilization usually internal. Fertilization usually internal. Are these points clear to all? The examples: Apis, Bombyx, Anopheles, Palaemonas. So Apis is the honeybee, Bombyx is the silkworm, Anopheles is the mosquito which carries Plasmodium, right, the female one. So Palaemonas is the scorpion, then Cancer or crab, the king crab, also known as Limulus. Limulus. King crab will be limulus, and this one is the living fossil. Living fossil means it has not underwent evolution ever since its origin. Then we also have another example called, we have another example called, wait guys, there is danger around. There is a wasp. Okay. So we have another example is Locusta. Another example is Locusta or locust. And this locust, this insect moves in huge groups. We call it gregarious. We call it gregarious. Quickly go through the examples, quickly. All right. Not a butterfly. I'm not scared of butterflies. See, it was a yellow wasp. All right. Moving on to the next, which is mollusca. Okay. Moving on to mollusca. Give me 1 second. Okay. The bumblebee. No, it was not the bumblebee. It was yellow wasp. Yes, bumblebee in cartoons looks like yellow wasp. Moving on to second largest phylum, which is Mollusca. Now listen, Mollusca, the second largest phylum, mostly marine, some freshwater and triploblastic, bilateral symmetry, coelomate. These are all true coelomates. Okay, soft unsegmented body. So the body is again unsegmented. See, we were writing the organisms which have unsegmented bodies. So another example of unsegmented here is mollusca. What is it? Mollusca, unsegmented. Echinodermata, unsegmented. Right? These are all unsegmented organisms. So I'm giving you many tables today alongside. So make sure you learn these, and I've already promised you to give a proper table of animal kingdom in my special class if you will learn them once. Is that clear? Okay. Is this point clear? Moving on to the next. Now the body is divided into head, muscular foot, and visceral hump. Clear? And the body is covered with a special covering known as mantle, and this mantle will secrete a shell made up of calcium carbonate, calcareous shell, which will be protective in nature. Now there is a space. See, this is how it looks like. This is the head, this is the visceral hump, and then this is the foot. So outside the here it releases this mantle. This is made up of calcium carbonate. This is the mantle. right? Now this space between the body and mental is called mental cavity. Clear? And in this mental cavity, space between the mental and body is known as the mental cavity. And in this mental cavity are present gills. Okay, where are these gills present? These gills are present in the mental cavity. Are these points clear to all? Please do genetics unit next. Harshita, I won't be able to do that. Genetics will come along, of course, but we are going to take units step by step. Okay, what next session? Morphology, anatomy, and structure organization, cockroach and frog as well. Now, feeding structure, very important feeding organ is radula. Circulatory structure is open. Excretion through nephridia, also through kidneys. Okay. Now, this radula is a feeding organ. It helps in cutting the food. So, we call it a rasping organ. Rasping means cutting. Rasping organ found in the mouth. Okay. Sexes are separate and these are the examples. Phyla, the apple snail. Unio, octopus, the devilfish. There are so many examples. The maximum examples are here. Devilfish, we have sepia, we have loligo, right? Then the dentalium. All those examples should be learned. Now I'm going to take a separate session where I am going to give you proper tricks also, like I've given you for fungi. So separate tricks for animal kingdom. So stay tuned. Okay. Now coming on to Echinodermata. See here. Echinodermata. Yes, please. Echinodermata, exclusively marine. Again, told you, exclusively marine. Triploblastic coelomates, endoskeleton made up of calcareous ossicles. Is that clear? Then, adults show radial symmetry, but larva shows bilateral symmetry. Very, very important. Very, very important. Otherwise, the same type of symmetry will be found in adults and the larva will show same symmetry. But in case of Echinodermata, adults have radial symmetry, larva has bilateral. So basically, larva is more advanced. Larva is more advanced because bilateral symmetry is considered to be more advanced feature. Are you getting these points? It has a unique thing, that is water vascular system. Now don't get confused. Write it separately in your notes at the top of it. Water canal system is in Porifera. Water vascular system is in Echinodermata. It has locomotion. It has special structures for locomotion known as tube feet. No excretory organs and no separate excretory organs. Excretion will occur through water vascular system itself. Sexes will be separate. Wait a second. Sexes will be separate. Okay, yes. And external fertilization will take place. Development would be indirect. Development would be indirect. Now the examples are, see, if you are solving PYQs, I'm sure you have solved PYQs, we'll solve them again also, but you have been solving PYQs. You have seen such questions that they ask scientific name, match it with the common name, right? So you have to learn both the names. Asterias is the starfish. Is a starfish. Echinus is the, quickly let me know what is Echinus. Quick. Yes, please. Starfish. What is Echinus? What is Antedon, Ophura, Cucumeria? Are you able to recollect? Quickly let me know. Vishwa, papules are also like structures present on the body. Papules are also present on the body of the starfish or the echinoderms. Let's stay within NCERT. This is where we are going to make huge mistake. Yes, Echinus is the sea urchin, right? Antedon. Who tells me about Antedon? Antedon. Who tells me about Antedon? Quickly. Cucumeria is easy to remember. Sea cucumber. है ना? Cucumeria is sea cucumber. Yes. Asterias starfish, cucumbers. Antedon is sea lily. Very good. Sea lily. Ophura is brittle star. Right? So these common names should be well learned. Right. All right, coming on to the last phylum of non-chordates, which is Hemichordata. Can anyone tell me which is one feature of Hemichordata which resembles with chordata? Why do we call them hemichordates, half chordates? What is that feature which resembles? resembles the chordates. Yes, feature of hemichordata that resembles the chordates. Anyone? Your paper is 100% clear if you know this. Stomochord. No. What is that chordate-like feature? No, 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 no. Chordate-like feature is pharyngeal gill slits. Pharyngeal gill slits is the chordate-like feature. This one feature is shared between chordates as well as hemichordates. Okay, earlier it was placed under chordates because of stomochord. Anyways, now these are exclusively marine. They are exclusively marine. Your homework is done. Exclusively marine. Bilateral, triploblastic, coelomates, body divided into three parts: proboscis, collar, and trunk. These are the three parts. Gills present. Respiration through gills is called branchial respiration. Now, earlier placed under chordates, but now separate. Circulatory system is open type. Hemichordates also have open type of circulatory system. Now we call them tongue worms because they look like tongue worms or acorn worms. Guys, stay patient, stay energetic, because I know it's already 7. We have almost completed 2 chapters, but plant kingdom is still coming up. And I have beautiful flowcharts for you. I've kept it for the last. Flowcharts will help you learn those chapters so easily. And then starting with chordata. Chordata. Okay. Now see. Chordata has these features. It has notochord, it has dorsal hollow nerve cord, it has pharyngeal gill slits, post-anal tail, and a thyroid gland. No doubt this is a feature, but it is out of NCERT. We'll skip it. So notochord, you can just skip it. Endostyle or thyroid gland. So notochord is solid. It is dorsal. And what is the origin? It is mesodermal always. Notochord is mesodermal. Nerve cord, dorsal, hollow, and single. And this is ectodermal. Pharyngeal gill slits, the unique feature, post-anal tail. Are you getting it? Then other features would be bilateral symmetry, triploblastic, coelomate, organ system, and closed circulatory system mostly. And then subphylum. It can be divided into subphylum. See, you must remember what is the taxon of Urochordata. It is a subphylum. Now, chordates have been divided into urochordates, cephalochordates, and vertebrates. Urochordates, they have notochord. If an organism has notochord even once in their lifetime, even for few minutes, it will be regarded as a chordate. But not just for few minutes, it has notochord during the entire larval stage. Is this clear? Acidia, salpa, herdmania, doliolum. These are all examples of what? These are all examples of Urochordata. Sorry, we have made this mistake. These are wrong. Okay, are these points clear? Yes. Doliolum. Clear? And also herdmania. Let me write it properly. Also known as tunicata. Why? Because they have tunic. Tunicata because they have tunic, a body cover made up of tunicin, which is a cellulose-like compound. Yes. Then cephalochordata. Session will be till 8 o'clock. I'll leave you 5-10 minutes before 8, but yes, we need this time. Notochord is present throughout life from head to tail. While in Urochordata, it is only present in the larval form and only in the tail region. While here, throughout life, it persists from head to tail. So this is being a cephalochordata. Example is Branchiostoma or Amphioxus. Now vertebrates, in this the notochord will change into, notochord becomes vertebral column. It gets modified into vertebral column. That is why the name is vertebrata. Now see, vertebrata and urochord, see, Urochordates and cephalochordates are called primitive chordates, cephalochordates. These are primitive chordates, or we call them protochordates, primitive or protochordates together, right? While this vertebrata is a modern chordate. So protochordates and vertebrates are the modern or the true chordates. Is this point clear? Or euchordates? Anyone with any doubt? Yes, it persists throughout the lifetime. Clear? Now, vertebral column present, notochord replaced by the vertebral column, well-developed brain, closed circulatory system, and paired sense organs will be present. So, see, I have already told you about urochordate. They are exclusively marine. Homework done. Exclusively marine. Body covered with test or tunic made up of tunicin, cellulose-like substance, told you. Body within the sac-like structure, unsegmented body. Notochord only in the larval tail, we have already discussed it. Absent in the adults, respire through gill slits, then open circulatory system. So chordate which has open circulatory system is this, Urochordata. Then pharyngeal gill slits, hermaphrodites, indirect development, tadpole-like larva, Acidia salpadolulum, then herdmania. All these are examples. Cephalochordates, marine, fish-like, elongated, notochord head to tail, persist throughout life. Rest is about notochord, nerve cord, which is similar. Gill slits, body segments are present, closed circulatory system. Okay. True coelomes, sexes are separate, external fertilization, indirect development. Example is branchiostoma. Then coming on to chordates. Now coming on to vertebrates. Now subphylum Vertebrata can be divided into two superclasses, right? One is Pisces, second will be superclass. Sorry. This is a bit of a mistake. Wait. So we have here cyclostoma. So we have agnathostoma here. This is agnathostoma. And second would be gnathostoma. Right? Agnathostoma means they will not have jaws. They are jawless. Gnathostoma will have jaws. Gnathostoma will be further divided into Pisces and Tetrapoda. Are you getting that? Yes. Right. Now is it clear? They will be further of two types, cartilaginous and bony. While Tetrapoda will be of four types. We'll talk about them one by one. Everyone is clear with what is being discussed. So these are jawless vertebrates, eel-like body without jaws. But if they don't have jaws, they have circular mouth. That is why the name is cyclostoma. Cyclo means circular, stoma means mouth. They have circular mouth. Are these points clear to all? Paired fins and scales are absent. Very important feature. The endoskeleton is cartilaginous, median nostril. How many gill slits are present? 6 to 15. They are mostly ectoparasites on fishes, NCERT. Suctorial, the circular mouth is suctorial with rasping tongue. They are marine and they migrate to fresh water for spawning. They live in seas, but they will go to the freshwater rivers and streams to lay their eggs. And after spawning, they will die within few days. So the eggs will hatch, larva will come out, become adult, and come back to the oceans. Are these points clear? All points clear? Yes. Notochord does not persist, right? Then, examples. What are you writing, Shri? Notochord does not persist throughout life and tunicates. Right, you're right. Now, example is petromyzin or myxine. This is a PYQ, by the way. The previous year question. Lamprey, has to be matched with petromyzin. Hagfish has to be marked with myxine. So the common names are important. I'm telling you again and again. By when the series will be completed? Very soon, before your exams, of course. Now coming on to Gnethostoma. We are going to talk about Gnethostoma, all the organisms which has jaws. So starting with first superclass, which is Pisces. We know Pisces fishes, these are true fishes. They are of two types: cartilaginous chondrichthyes, right? And osteichthyes, the bony ones. So chondrichthyes are marine, streamlined body, endoskeleton, cartilaginous. That is why they are cartilaginous. Mouth is ventral. Think of shark. Think of shark when you talk about cartilaginous fishes. Mouth is on the lower side, ventral, right? Placoid scales, 5 to 7 gill slits. Gill slits not covered by operculum. So it is open. Air bladder absent. So you will never see a shark static. So it has to keep swimming, or it will sink and die. Keep swimming constantly. Why? Because it does not have a air bladder. Heterocircle tail. Then heterocircle tail is, see, this is how shark is. The tail has one the two lobes of the tail fin, they are of different size, longer and smaller. Are these points clear to everyone? Please let me know. Please let me know that these points are clear to everyone. Yes, please. Please let me know these points are clear to everyone. Clear? Perfect. Moving on then. So the example, see, the teeth. We always see backwardly directed teeth of shark, right? So these backwardly directed teeth are modified placoid scales. These are backwardly directed. You will always see these cuts. So there is no operculum, remember? So this is how a shark looks like. And internal fertilization, and they give birth. So most of the species are viviparous. They give birth to the young one. Examples: Scoliodon, dogfish; Pristis is sawfish; Carcodon is a great white shark; and Trigon is a stingray. Points clear to everyone? Please let me know. Guys, there is some electricity issue. I won't be visible, but the screen will be visible. Just keep listening to my voice and keep following. Now, coming on to osteichthyes. Osteichthyes are the bony fishes. They occur in marine as well as freshwater. Endoskeleton is made up of bones. Body is streamlined. Mouth is terminal. Mouth is terminal. 4 pairs of gills. How many gills were present here? 5 to 7 pairs. And how many pairs are here? 4 pairs. And now they have operculum. The scales are of cycloid and ctenoid type. Air bladder present. So now you will see these fishes static at one place. Is it clear? The tail is usually homocircle. In few it can be heterocircle. Okay. Then the external fertilization mostly, and most of them are egg laying, so oviparous. Now see the examples: labio, cartla, clarius, exocetus, hippocampus is a seahorse. Now, why do we call flying fish as a flying fish? It has a very large pectoral fin. So it glides beautifully outside the water, pectoral fin. Okay, are these points clear to everyone? All right, moving on to the classes of Tetrapoda. See, first Tetrapoda is amphibian. As the name says, it has dual habitat, lives in water as well as on land. Cold-blooded. Fishes are also cold-blooded. All the Pisces are also cold-blooded. Cold-blooded means they cannot regulate the body temperature. They have moist skin and without scales. No scales are present on the skin. Moist skin is going to help. Moist skin is going to help in cutaneous respiration. Cutaneous respiration. Are these points clear? 2 pairs of limbs, digits are without claws, respiration through gills in the larval stage, and lungs and skin in case of adult. Is that clear? Tadpole tadpole is the larva, is going to respire through gills. Adult is going to respire through three things. It's going to respire through three things: lungs, mouth, and skin. So through skin it respires when it is underwater, and lungs and mouth are used on land. So this is how it respires. Three-chambered heart is present. External fertilization usually lays eggs, so it is oviparous, and indirect development because it has a larval stage known as tadpole. Rana tigrina, the frog, hyla, the tree frog. Then we have bufo, the toad, salamander, ichthyopsis, which is fish-like. Ichthi word is for fish, fish-like amphibian. Is this point clear? Clear? Salamandra looks like lizard. So if you have to identify on the basis of the picture, Salamandra looks like the reptile, but are these points clear to everyone? Right. Then mostly terrestrial. Lungs for respiration. Heart is three-chambered. Fishes have two-chambered heart. Remember, fishes have how many? Two-chambered heart. Fishes, two-chambered. Amphibians and reptiles have three-chambered. Exception is crocodiles, alligator, right, ghadial, and then birds and mammals. They have four-chambered heart. Crocodile is a reptile, but it has a four-chambered heart. Internal respiration, mostly oviparous, means it is going to lay eggs, and the eggs have tough shell around it for protection. Are these points clear? Okay, moving on to the next. Now we have next. Limbs are present in most, but they are absent in snakes. So these are the examples you must remember: Keelone, turtle, testudo, tortoise, chameleon, the girgid, the garden lizard is Calotis, crocodilus, naja is the, and then crate, and then vipera. Now another homework, you have to find out which of these are the poisonous snakes. Yes, can you name the poisonous snakes here? You have to separate which one of these are the poisonous snakes. Very good, Shri. You should all be busy writing down the notes, not in chatting, which is very good. Which is very good. Right, Prajwal, single circulation. Which one is the poisonous snake? This is your homework, clearly given. Naja, vipera, yes. Then crate. These are all poisonous snakes. Right? Then coming on to the aves or the birds. So talking about birds here. Okay, coming on to birds. Birds are the first warm-blooded organisms, endothermal. Body temperature will stay constant. Body covered with feathers. Their forelimbs are modified into wings. Hind limbs will be used for walking and swimming. Right? Then or for perching. Perching means holding onto the tree or the right. Then beak is present for biting, but teeth are absent. Light-weighted bones, hollow bones. These hollow bones is one adaptation for flight. We call hollow bones as pneumatic bones. Our bones are filled with marrow. These bones are solid, filled with air to keep their body light for flying. Four-chambered heart, lungs with air sacs, egg-laying oviparous, and they have hard calcareous shells. So these are the examples. Crow is corvus, columba is pigeon, psittacula, parrot, your national, sorry, not the national bird. National bird is peacock, the pavo, right? Struthio is ostrich, the largest cell. The egg of ostrich is the largest cell. Penguin is the Ctenoditis, and Neophron is the vulture. Are these points clear to all? And now moving on to Mammalia, warm-blooded. Quickly, warm-blooded, body covered with hair, mammary glands present, external ears present, two pairs of limbs, right? We have two arms, two legs, four-chambered heart, diaphragm, very unique feature. Diaphragm is only found in mammals, right? Now we have heterodont dentition. We have different types of teeth. Our teeth appear twice in the lifetime, diphyodont, and we have thecodont teeth. They are present in the cavity, jaw cavity. Internal fertilization, mostly viviparous except for monotremes. Monotremes are egg-laying mammals. Monotremes are the egg-laying mammals, platypus or echidna. Monotremes are the egg-laying mammals. Is that clear? Rest, macropus, pteropus, this is a pouched mammal. Kangaroo. Pteropus, flying fox. Camellias is a camel. Macaca is monkey. Homo sapiens, us. Is that clear? We are all placental mammals. Are these points clear to all? Anyone with any doubt, please let me know. Now, then, Belinotera and Elephas. Belinotera is the blue whale, the biggest animal, right? And Elephas is the elephant. Now, very, very important, you often see sometimes that which of the following character is present in a particular phylum or class without any exception. Please see. In mammals, mammary glands, hair at some stage, 3-ear ossicles, chalo, this is not really in your syllabus, you can skip this, diaphragm and 4-chambered heart. These characteristics are always present in mammals without exception. You get this question often. But viviparity may or may not be present. External ear pinna may or may not be present. Teeth may be absent in some, like in whales, and placenta may or may not be present. So this is one very important NEET bonus point that I am giving you. Are these points clear to everyone? And once again, guys, those of you who have joined me late, please make sure set your alarm for 19th April 2 to 5 p.m. You have to take these mock tests every day. Enroll for free because see the benefits are you will get all India rank. Top 50 will get mentorship after every test. You will understand how to improve, what mistakes you made, what things you should have taken care of. Top 10 will get WhatsApp guidance and top 3 500 rupees voucher. But here the most important thing is mentorship, which I feel. Now the next chapter that we are going to start, guys, is a plant kingdom. I'm going to skip living world and leave it for the last. Let's start with the plant kingdom. Very, very important topic. Now this whole chapter will be full of flowcharts. You may not be able to write all of this with me. So I will of course be sharing the PDF. Please understand these things now. Is that clear? Please understand. So first of all, the types of classification given in your NCERT, the different types, natural systems. See, the different types of classification are natural system. Natural system classification was given by Bentham and Hooker. Okay, Bentham and Hooker. And it was based on many characters like morphology, anatomy, biochemistry, cytology, phylogeny, embryology, right? Then ultrastructure and phytochemistry. Now, basically, because this natural system of classification was based on so many bases, so it is, of course, more authentic. While artificial system of classification given by Aristotle and Linnaeus was just based on superficial criteria. It was based on superficial criteria, so it is not very authentic. What was the basis of artificial classification? You need to remember these bases. I'm telling you again and again. Take it as a homework. See, in this session, we have done multiple classifications, right? Make sure that you note down the basis of each and every classification. Basis of classification of 2 kingdom, 3 kingdom, 4 kingdom, 5 kingdom, 6 kingdom. Then in fungi, we've seen the classification was based on 3 factors, right? Then protozoans classification, and now these classification. So you must know what were the bases. And in animal kingdom also there are various bases. So artificial system was less authentic because it was based on superficial criteria like habit, habitat, color, shape, and so on. Clear? It was given by Aristotle and Linnaeus. Then we have chemotaxonomy. Chemotaxonomy is based on the chemical composition. Cytotaxonomy. Cytotaxonomy is based on the cell structure, the chromosome number. That is cytotaxonomy. Numerical taxonomy. Numerical system of taxonomy is based on hundreds of criteria. So basically, numerical taxonomy uses computers, right? So actually, every character is given a code, and now this computer helps us compare those different codes. So numerical taxonomy is also very authentic. So here, the main feature is Every character has a code. Every character has a code, and we can use hundreds of characters to classify the organisms. Hundreds of characters can be used because it is not humanly done. It is done by the computers. Then phylogenetic system. Phylogenetic system is given by Engler and Prandtl. This is more scientific and more natural method, and since the name says phylogenetic, it considers the organism with common ancestry should be kept in one taxa. So what is the underlining statement of phylogenetic? Those which have common origin, keep them in one category. Is that clear? These are different types of classifications. Is this point clear to all? Quickly, let me know. Give me a thumbs up that you're all studying with full energy. See, it's last 20 minutes and we have to complete this plant kingdom. Quickly let me know. Clear? Perfect. Now, the three different types of life cycles that you will see in the plant kingdom are haplontic life cycle, haplodiplontic life cycle, and diplontic life cycle. Now see, diplontic life cycle will be seen in gymnosperms, angiosperms, and few fungi. And which fungi? Fucus. Haplodiplontic is seen in bryophytes and pteridophytes, while haplontic life cycle is only seen in algae. It is only seen in algae. Are these points clear? Algae, this is bryophytes and pteridophytes, this is gymnosperms and algae. Now see, algae is going to show all types of life cycles. Mostly, see, if the question is algae shows which type of life cycle, answer should be haplontic. But only haplontic? No. Algae shows haplontic life cycle. Algae shows haplodiplontic. Now the examples of algae which show haplodiplontic are Ectocarpus, Polysiphonia and Kelps. Examples are super important here. And the algae which shows diplontic life cycle is Fucus. Are these points clear to everyone? Please let me know. Please let me know. Yes, please. Are these points clear to everyone? Perfect. See, I'll be sharing all these flowcharts with you, but please stay very clear, energetic and attentive to understand the flowcharts. All right, moving on to the next. Now this is the first life cycle that I'm going to talk about, that is haplontic life cycle in algae. What is haplontic? Means the major life cycle will have haploid stages. Now haplontic life cycle always starts with the parents which are haploid. Is that clear? So in haplontic life cycle, the parents will be of, okay, parents will be haploid in nature. So the haploid body is always known as gametophyte. Please understand certain terms. I'm sure there are few of you who have never been clear with these terms. What is sporophyte? What is gametophyte? Or haplontic or diplontic? Let's get it sorted for once and for all. Number one, haplontic life cycle means most of the time in their life cycle will be haploid in nature. In such cases, parents are always haploid, means single set of chromosomes. Now, the ones which have haploid set of chromosomes, that body is also known as gametophyte. And gametophyte is always going to produce gametes. Are these points clear? They are always going to produce gametes. There are certain things. Be very thorough with it. What? Sorry. That is haplontic means major life cycle would be haploid, right? Diplontic means major life cycle would be 2n. Haplodiplontic means major life cycle, haplodiplontic would be, it will have n also, and for sufficient time it will have diploid phase also. Second, gametophyte. Whenever we use the word gametophyte, it means it is haploid in nature. Whenever we say it is sporophyte, it means it will be 2n in nature. Are these points clear? Second point. Third point, sorry. Sporogametophyte will always produce gametes. It will always produce gametes. Sporophyte will always produce spores. See, I know some of you are struggling with certain things and you might feel, okay, is it too late? No, there are certain things we can still uh, cover up. Okay, now listen. So In haplontic life cycle of algae, parents are N gametophyte, and since it is a gametophyte, it is going to produce gametes, right? See here, it is going to produce gametes. So who will produce gametes? Sex organs. So parents will have sex organs being a gametophyte because it is going to produce gametes, right? Now please let me tell you, and you tell me, use your common sense. If the parent is haploid and gametes are always haploid, which kind of cell division will take place to produce gametes? Mitosis. N to N, it is mitosis. So parents will produce gametes by mitosis, and these gametes will undergo fertilization or syngamy to produce a zygote. Now this zygote is 2N, and this is the only phase which is diploid. Now quickly the zygote is going to divide by meiosis to form spores. Now why do we call this a haplontic phase? Because here the zygote is very short-lived. The 2N phase is super short-lived. Are you getting that? And these spores will undergo mitosis or germination to produce parents again. Is that clear? Now, see, this is the evolution. First came the algae, then came bryophytes, then came pteridophytes, then came gymnosperms and angiosperms, right? And this is the life cycle. Algae shows, I told you, all the life cycles. These two are haplodiploidic and these two are diploidic. Clear? Now, plants can be divided in this form also. There are plants which will bear seeds and which will not bear seeds. Without seeds, plants are known as cryptogams. With seeds, plants will be known as phanerogams. Are these points clear? Now, the cryptogams, that means who do not produce seeds, they can be further differentiated. Not differentiated body or undifferentiated body, that means no true root, stem or leaf, right? They will be algae or bryophytes. But then no seeds but well differentiated body, right? These will be pteridophytes. See here. Now in algae and bryophytes, vascular system is also absent. But in pteridophytes, body has true root, true stem, true leaf. Also it has vascular system also. On the other side, we have seed-bearing plants. Seed-bearing plants means those which are called phanerogams, right? They will have vascular tissue. Body will be well differentiated. Now they can be further divided on the basis whether they produce naked seeds or the seeds are enclosed within the fruit. So naked seeds are present in gymnosperms. Seeds are enclosed within the fruit in case of angiosperms. Are these points clear to everyone? Are these points clear to everyone? Okay. So coming on to algae first of all. So algae, see here, algae, what type of body is present in algae? Algae has simple thalloid body. Thalloid means body will not be differentiated into true root, stem and leaf. Okay. No vascular system, no seeds, no flowers. Habitat, mostly aquatic. Some of them will be found on moist soil, stones and wood also. Size will be variable. They can be microscopic like volvox and eulothrix. They can be 100 meters tall also, like in case of kelps. So microscopic will be unicellular or they can be multicellular also. Unicellular we are talking about Chlamydomonas, chlorella, spirulina. This spirulina is also a single cell protein. It is a source of protein. These days people eat spirulina powder for the protein instead of protein powders. They are eating spirulina. Then habits. All algae are photosynthetic. All algae are autotrophic. Now, certain algae show symbiotic association also. Certain algae shows symbiotic association as well. Wait, let me use I'll use this highlighter. So certain algae they show symbiotic association, lichens, like algae and fungi. Then some algae are present in the body of sloth bear also. Again, symbiotic association. Are you getting these points clear? Profusely branched, Vishwa, means highly branched. Profuse means highly. Now, they have different life forms. Some algae are colonial. The example is Volvox, Filamentous, Eulothrix, and Spirogyra. And massive forms, example is kelps. See, this one slide is very, very important. Very, very important slide. Okay. Now coming on to the types of reproduction. Types of reproduction in algae. See here. Reproduction, wait. In algae is of three different types. You can see You can see asexual in the form of asexual spores, and the most common spores are zygospores, zoospores. Zoospore means they are motile, and eplanospores. See, this is a general term. In fungi also we have read that motile will be known as zoospores and non-motile will be known as eplanospores. Are these clear? Then vegetative reproduction will be by means of fragmentation. You cut down the algae, each fragment will give rise to new algae. Yes, you will get all these slides PDF. Clear? Now, fragmentation in Eulothrix, Spirogyra, they are filamentous. You break any filament, every filament will give rise to the whole new algae. Sexual reproduction. Now, see, they are all bisexual. First of all, this is 2022 ka question. Algae, the sexuality of algae is bisexual. It is monoecious. Now, in sexual reproduction, you will see gamete formation. Gametes can be of three different types. It can be homogamous, it can be anisogamous, and it can be oogamous. See, anisogamous, also known as heterogamy, isogamy, also known as homogamy. Are these points clear to all? Please let me know. Isogamy means both male female gamete will be alike. You cannot differentiate. And iso means they will be dissimilar. You can differentiate. Okay, this one is male, this one is female. And then oogamy, a special type of an isogamy in which the female gamete is large and non-motile. Very important. Oogamy is female gamete is large and non-motile, and male gamete is motile and small. Because motile, it will have flagella. Is this point clear? Is that clear? Now, one very, very important slide. Very, very important slide. See here. Economic importance. They are oxygen. They are oxygen producers. They are oxygen producers. Are these clear? They are main oxygen producers. They are main producers of food for aquatic food chain. Then this is a PYQ, hydrocolloids. They have water holding capacity. So they are water holding substances. Algin and carrageen are the hydrocolloids. Algin is obtained from the brown algae and carrageen is obtained from the red algae. This is a PYQ. Now how to remember? AB. Algin from brown algae, carrageen from red algae. ABCR. Is that clear? Now, why do we use hydrocolloids? For transport of substances, seedlings, and plants from one place to another in order to prevent desiccation, dehydration. They are very important food sources. Again, a PYQ. Sargassum, Laminaria, Chlorella, Spirulina. See, in these notes, you will get all the PYQs mentioned here. Chlorella is used as a space food. Very important question. Also, you know that it is a protein source. So, chlorella and spirulina, they are both used as protein sources, single cell protein. Now, we also get agar, which makes our ice cream actually gooey, right? So, agar is used in ice creams, jellies, and culture media and in cosmetics. So, what are the sources? The sources are Gracilaria and Gelidium. Both are red algae. So, the sources of agar are red algae. Are these clear? Yeah. No problem, Shri. You can take these notes and revise. I would recommend don't make notes for plant kingdom. Just use them as such. They are sufficiently colorful. They are flowcharts. You don't need to make the flowcharts here. Okay. Just keep understanding every point. Now listen. We have three types of algae: chlorophyce, phaeophyce, and rhodophyce. You have to remember, and the way to remember is here. See, for mnemonic for phaeophyces, ecto helps Sargon learn dicot function. So ecto is ectocarpus, helps. From helps you have to remember kelps. Sargon is sargassum, learn is laminaria, dicot is dictyota, and function is fucus. Okay, will you be able to learn? For red algae, we have P2, G2. P2 is Porifera and Polysiphonia, and then G2 is Gelidium and Gracilaria. Clear? And rest all the examples that are given in NCERT belong to green algae. Green algae is bright green and dark green. Brown algae is olive green to brown, and red algae is red in color. Now the pigments, very important. Pigments present. See, this table is super important. Chlorophyll. See, chlorophyll A is present in all. Chlorophyll A is present in all. Write down A. Okay, and then B, C, D in order. First, learn the order C, P, R. As a doctor, you must remember CPR, right? Then CPR on top, and then A, B, C, D in order. Apart from A and C, Phaeophyce also has fucoxanthin. Rhodophyce, apart from A and D, has phycoerythrin. Now, fucoxanthin is a xanthophil and it has carotenoids also. Are these points clear to everyone? Stored form of food, again, very important. Now, these tables are very, very important. Stored form of food is starch and oil droplets. Here we have mannitol, and in Rhodophyce we have Floridian starch. And the structure of Floridian starch is similar to amylopectin and glycogen. These are all important points. Talking about the cell wall, every algae has cellulose. Every algae has cellulose. But Phaeophyce will have elgin also in the outer layer. Cellulose will be on the inner side. And Rhodophyce will have pectin and polysulfate esters on the outer side. Inner will be cellulose. So cellulose is common. Now talking about the number of flagella. In C, that is Chlorophyce, the number of flagella are 2 to 8. Both are equal and on tip. Can you see that? In Phaeophyce, we have again two flagella, but these two flagella are unequal and they are lateral, not apical. They are on the side. One will be long, another will be short. And Rhodophyce, there will be no flagella. Are these points clear? Super slides, Anna? Yes, I know these are super slides because it took me a lot of time to make. I am trying to make things easy for you, and similarly in all other classes also you will get these notes for your quick revision. Make sure that you don't miss out these classes, okay? And we'll be discussing the questions in the special classes. Now thallus is unicellular or multicellular. It can be colonial or filamentous. Phaeophyce only has multicellular, and Rhodophyce also only has multicellular. In Phaeophyce, multicellular, thallus has three parts. See, this is a Phaeophyce. So this part is called holdfast. This stem-like structure is known as stipe, and this leaf-like structure will be known as frond. Is that clear? Holdfast, stipe, and frond. While the structure of Rhodophyce is very complex, very plant-like. Is this point clear? Talking about the habitat, mostly fresh or brackish, mostly marine and rarely freshwater, mostly marine and few freshwater. So just take care of these words rarely and few because NCERT has used different words almost having the similar meanings. But when it is written rarely, it is Phaeophyce. When it is written few, it is Rhodophyce. So don't combine rarely with R. Okay, just a trick to remember. Clear? Class is almost going to be over. Wait a second. So this is the types of reproductions. We've already discussed with you. Fragmentation will be common to all. Asexual spores will be of different type. Here we have zoospores, and in asexual, we don't have zoospores. There are non-motile spores, so we call them eplanospores. In sexual reproduction, aniso, and oogamous. Phaeophyce also shows all the three types, iso, and oogamous, but Rhodophyce will only show oogamous type of reproduction. Are these points clear? So which one has isogamy and anisogamy? Which one has motile spores and non-motile spores? Download the notes and learn them very well. Is that point clear? Now, moving on to just a second. Okay. Yes, please. Moving on to, wait a second, guys. Yes. Can I extend the class? Message कर देना. Chalo. Let's move on, guys. So we were talking about, okay, we were talking about bryophytes, right? Bryophytes are known as amphibians of plant kingdom because they need water to complete their life cycle, and they're found in the moist, shady places, damp, humid. So see, moisture, dampness is nearby. Why? Because they need water for completing their life cycle. So here we get assertion, reason, question, yes or no? Now, another very important slide, features of bryophytes. The main plant body will be haplontic, haploid. See, Bryophytes have haplodiplontic life cycle. है ना? Haplodiplontic life cycle. Why? Because the main plant body will be, or the dominant phase will be haploid, but diplontic phase will also get sufficient time. Okay, the life cycle is haplodiplontic. Sporophytic body will be attached to the gametophyte for nutrition. Sporophyte will not have independent existence. Rather, the body will be divided into three parts: foot, seta, and capsule. The body is not differentiated into true leaf, stem, and root, so it has a thyloid body. But but still it is more differentiated than algae. Okay, they are amphibians, we already know, but body is not differentiated into true root, stem, and leaf, but still more differentiated than algae. So it has root-like structures called rhizoids, stem-like structures, leaf-like structures. Are these points clear to everyone? Very important slide. Very important slide. Economic importance of bryophytes. Pioneer species at multiple places. Pioneer species means where there was no soil, these bryophytes have the capacity to form soil. So lichens and mosses, they are pioneer species. Okay, they play important role in succession of bare rocks. They prevent soil erosion. So this is their ecological importance because they form a dense mat. They form a dense mat. Is this point clear? Okay, then they can be used as food for herbaceous mammals and birds. They are used as fuel. Sphagnum is the example, and it is a PYQ. It is a peat moss, and it is also used as a packaging material. Like algae was used as a hydrocolloid, right? Similarly, these can be used as packaging material because they also have water holding capacity. Are these points clear to everyone? Are these points clear to everyone? Quickly let me know. Now, this is the life cycle. See here. So see, first of all, uh, we see here that the main body, the parents are haploid in nature. The parent body is haploid. Haploid is also known as gametophyte. So the dominant phase is haploid. And the parents, see, how are the parents? Just remember your parents. Your parents are always dominating. So this is a trick to remember. Okay, so parents are always dominating. Parents will always make their own food. They are autotrophic. They're always independent. So these are the characters of parents. No matter what the generation is, whether it is haplontic or diplontic, parents will always have these three characteristics, right? Parents will always have these three characteristics. What are the characteristics? They are autotrophic, they are dominating, they are independent. So whatever you are studying, when we talk about parents, they must have these features. Is this point clear? Is this point clear to all? Right? Okay. And the body, as I told you, body is not differentiated into true root, stem, and leaf, but still differentiated more than algae. So it has root-like rhizoids, stem-like main axis, leaf-like leaf structures. Now see, it is a gametophyte. I told you, gametophyte is always going to bear gametes. And gametes will be present, they will be produced within the sex organs. Yes? So there are two types of sex organs here. Remember, antheridium and archegonium. Archegonium is flask-shaped, right? This is a picture of the archegonium and antheridium. So these sex organs are jacketed. They have a covering. Do you see there is this layer of cells that I have made here? This layer of cells. These are the cells which form a jacket, a cover around it. Is that clear? So now antheridium is an organ in which male gametes will be produced, and archegonium is going to produce the female gametes. Yes, female gamete is known as egg, male gamete is known as entherozoid. This is how entherozoid looks like. So it is biflagellated. Entherozoid is biflagellated. This is all given in your NCERT. This is all given in your NCERT. I have summarized everything in these flowcharts. Clear? Egg is always non-motile, be it our egg or this egg. Egg is always non-motile. Now the two gametes are going to undergo fertilization and form a structure known as zygote. This is the first 2N cell, and 2N is known as sporophytic generation, right? 2N is a sporophytic generation. So zygote is the first cell of sporophytic generation. So give me thumbs up that all of you are liking these notes and you will be downloading these notes and learning them quickly. Give me a thumbs up. Quickly give me a thumbs up that these things are very, very clear to all of you, right? Now, sporophyte. This is a sporophyte. Now, see, zygote will undergo mitosis. It will form a multicellular body known as sporophyte, and it will have foot, seta, and capsule. So, this is how it looks. Capsule will be a swollen structure inside which spores will be produced. The spores are produced by meiosis, and meiosis will give rise to haploid structure. So spores are haploid. So the first cell of gametophytic generation is spore because it is haploid. These will germinate to again produce haploid parents. Please let me know. Are these slides useful? Right? So make use of them, crack your exam and let me know. Okay? Chalo. Moving on to now, bryophytes are of two types. We have read that bryophytes are of two types. One are liverworts, second are mosses. So, in liverworts, we have two examples, Rixia and we have Markensia. We have two examples, Rixia and Markensia. In mosses, we have three examples, Sphagnums, Funaria and Polytrichum. Clear? Now, the difference is basically how the thallus looks like and also in their gametophytic generation. See, liverworts will have a flattened body, dorsoventrally flattened, as if you have just pressed it against the soil. See, dorsoventrally flattened body. On the lower side, we have these root-like structures called rhizoids, right? Now, they are pressed to the substratum. Now, see, if we talk about Markensia, it is unisexual. So, male and female thallus will be separate, while Rixia is bisexual. So, male and female thallus will be similar. Okay, so on the same body it will have both the sex organs. Now if I talk about Markensia, so for example it will have this stem like structure known as archegoniophore which will bear archegonia. Okay, so this is a female thallus. If I talk about the male thallus will have enthridiophore. It will have enthridiophore and it will bear enthridium. This will be enthridium. Are these points clear? So these structures are cup-shaped structures known as gamete cups and they will form gamete. These gamete are the asexual buds. They are multicellular, green and they are buds, so they help in asexual reproduction. Are these points clear? While enthridium is the male sex organ, male sex organ, archegonia is the female sex organ. Please let me know that these points are clear. Super clear? Now, if I talk about mosses, now what is the difference? So, mosses have a special stage known as protonema stage. It has a special stage known as protonema stage, right? And protonema stage is a creeping stage. So, first of all, there will be a spore, okay? And spore will form a protonema stage, and protonema stage will give rise to a leafy stage. So, you must know the differences between the two. See, so these are the examples that which liverwort is monoecious and which one is dioecious, just a bonus point. So this is the life cycle. Let's first see the life cycle of a moss. So parent, let's again talk about parents. How are parents? Parents are always dominating, they are free living, they are autotrophic, right? They are independent. Clear? Now, the parent is in the protonema stage. So what type of body does the parent have? It has a leafy body. Sorry, it has a creeping body. I am talking about moss. I'm talking about moss. So the spore will give rise to the creeping body which is known as protonema stage. This stage will then give rise to a leafy stage. Both these stages are going to form the gametophytic generation. Both these are both gametophyte. These are parent bodies. These both represent the gametophyte. Point clear to all. Please let me know. So protonema stage directly develops from the spore. This is important. Creeping, green, filamentous, and it gives rise to a bud. And this bud will produce a leafy stage. Is that clear? While the protonema is going to crawl on the ground, this leafy stage is going to grow vertically upwards. It is upright. Is that clear? Now this leafy stage is going to produce sex organs. Again, the same that we have read, Antheridium and Archegonium. They are going to produce gametes, nothing different. The only difference between liverwort and moss in the life cycle is this stage, this part. Is that clear? The gametophytic stage. Here it has protonema and leafy stage. Please let me know that these points are clear. Rest everything is same. And another point to remember is that mosses have elaborate spore dispersal mechanism. Mosses have elaborate spore dispersal mechanism. This is another point to be taken. Are these points clear to everyone? Now see, next coming on to pteridophytes. Pteridophytes are further divided. See, pteridophytes are the first terrestrial plants. Now the dependence on water will not entirely go, but it will reduce a lot. So now these are the first land plants, which are pteridophytes. We have 4 classes in here, and which one are Psilopsida, Lycopsida, also known as club moss, Sphinopsida, the horsetails, Pteropsida, the ferns. Learn the examples. Psilopsida, easy to remember, Xylotum. Lycopsida, Slegionella and Lycopodium. The 2 examples here. Sphinopsida, Equisetum. Pteropsida, Pteris, Dryoteris, Ediantum. You have gotten these examples in your questions as match the following. So make sure that you learn them very well. It actually took me a lot of time to make these slides, right? So make sure that you use them. I will love it if they are helpful to you. Now, life cycle of Pteridophyta. See, these are the first terrestrial plants I told you. Again, it has haplodiplontic life cycle. So there will be sufficient haplontic cycle, sufficiently diplontic life cycle also. Sorry, diplontic phase also, not life cycle. So life cycle is haplodiplontic. But the difference now from bryophytes is bryophytes has dominant phase as haplontic phase. The parents of bryophytes are haplontic. But in pteridophytes, the parents are going to be sporophytic. Are you getting that? They will be diploid. Again, parents are dominanting, free living, autotrophic, independent. These are general characters of all parents in the world, right? They are dominating, free living, autotrophic, independent. But here you have to remember, unlike bryophytes, the parents of pteridophytes are sporophytic. Clear? Now, sporophyte means they're going to produce spores. They're going to produce spores. See, they're going to produce spores. But now spores are produced in certain structures known as sporangia. And there are certain special leaves in which sporangia are present. Those leaves are known as sporophylls. See here. So parents, body is sporophytic, multicellular, and now for the first time the body will be well differentiated into true leaf, true root, true stem. Is that clear? Well developed vascular system will also be found. Now there are some small sized leaves, microphylls. If it has large sized leaves, macrophylls. Which one has large sized leaves? Ferns. Small sized leaves will be found in Slegionella, Sanjeevani booty, which Hanumanji went for, right? To revive Lakshmana. That was Slegionella. Is that clear? Now, since they have leaves, so there are some special spore-bearing leaves. These spore-bearing leaves are known as sporophylls. Clear? These sporophylls they may aggregate to form cone, but it may or may not be present. In pteridophytes, cones may or may not be present. Now, which ones form cones are Equisetum and Slegionella? These form cones. Are these points clear to all? Then sporangia. Leaves will have special structures called sporangia, and there are cells called spore mother cells, which will undergo meiosis to form spores, the first cell of gametophytic generation. Now all spores are similar except in Slegionella and Salvinia. Now here in Slegionella and Salvinia, male-female spores are different. So we call heterospory has begun here. If male-female spores are identical, we call it homosporous. Algae, bryophytes, pteridophytes are all homosporous. Pteridophytes are also mostly homosporous. See, I've written here, mostly homosporous. Clear? Now, spores will germinate to give rise to the undifferentiated body known as prothallus, which is gametophytic. Gametophyte will bear sex organs, again the same sex organs, antheridium and archegonia, right? They will again form antherozoids and egg cells. and they will fuse to form zygote 2n, and zygote will develop into parent. Are these points clear? Are these points clear to all? Now this is going to be the last one to have Enthredia. This is going to have Enthredia. Are these points clear to everyone? Anyone with any doubt, please let me know. Clear? Okay. So these are the features of gametophyte. So see, this is a table telling you which all are homosporous and which one are heterosporous. Clear? Moving on to the next, very important NCERT line. Evolution of seed habitat happened in pteridophytes. See, the development of zygotes into young embryo take place in female gametophytes, and this is the beginning of seed habit. This is the beginning of seed habit. Are these points clear? So seed habit evolved in pteridophytes. Points clear? Anyone with any doubt, please let me know. So these are the pictures that you must remember. And talking about gymnosperms now. Gymnosperms are plants with naked seeds because the ovules are not enclosed by any ovary and they remain exposed. Is that clear? So seeds are of course post for fertilization, but they are not covered, hence naked. So gymnosperms will have medium-sized trees also, tall trees also, shrubs also. A giant redwood tree or sequoia is the tallest tree species. Now these gymnosperms, see, gymnosperms show some symbiotic association also. Okay, symbiotic association. For example, see here, in fungal association with roots of pinus will form mycorrhiza, while with cycus, cyanobacteria, nitrogen fixing cyanobacteria will form coralloid roots. So the trick to remember is PM. In pinus, it will form mycorrhiza, and triple C. That means in cycus, cyanobacteria will be present and it will form coralloid roots. Will you be able to remember these things? Please let me know. Then the stem can be unbranched in cycas and branched in pinus and citrus. Leaves can be simple as well as compound. If they're compound, they are pinnately compound. In cycas, they persist for years. They are well adapted to withstand extreme temperature conditions. In conifers, the needle-like leaves reduce the surface area, and thick cuticles, sunken stomata, they reduce the water loss. These are all adaptations to reduce the water loss. Please let me know that these points are clear to all of you. Thank you, Naman. Thank you. Is that point clear to everyone? So again told you this table is super important. Be it pigments, be it foods, these have appeared in many papers. Is that clear? So I am leaving you with these tables. And now, before we go, let's quickly revise. Let's quickly revise living world. Okay. See, in living world, the things that you must actually take care of. Number 1 point. Quickly, let's revise. What are the features of living beings? Metabolism, reproduction, structural growth, and then irritability. Now, out of this, the defining features. Let's talk about the defining features. The defining features are which are always present in all living beings. Number 1 is metabolism. There are 3 features which are defining metabolism, consciousness, and structural organization, cellular organization, without which no organism can be known as living. Are you getting that? These are the defining features. And the non-defining features would be growth and reproduction. Is that clear? Why growth? Because even non-living organisms grow. Living beings grow intrinsically and non-living beings grow extrinsically by absorbing substances from the outer side. Please let me know that these points are clear. Are these points clear to all? Okay. Then coming on to See, in unicellular organisms, growth and reproduction are not distinct. But in multicellular organisms, growth and reproduction will be separate phenomena. Reproduction, why do we call reproduction as non-defining? Because it will not be shown by all organisms like infertile couple, hybrids, honeybee. These will not reproduce. Are you getting that? Points clear to everyone? Now, similarly, see, we know about metabolism. In metabolism, one very important line of NCERT is that isolated metabolic reactions. See, what are metabolic reactions? All the chemical reactions going in our body. But if the same reactions occur in a test tube, they will neither be living nor be non-living. One very important line of NCERT. Are these clear? Then structural organization means all bodies should be made up of cells. Without being made up of cell, we will not call it living. Are these points clear? Okay. Then talking about diversity, as NCERT says, according to IUCN, you should know that there are around 1.7 million species, right? Rough estimate is 200 million species, which we talk about in ecology. And then Robert May said there are 7 million species. Yes. Then what is taxonomy? This term was given by Carlos Linnaeus. Taxonomy has 4 steps and you have to remember them in this order. The sequence. First is categorization. Second is identification. Third is classification followed by naming. Naming is the last point. Naming is last thing, nomenclature. Now, if I include phylogenetic study also, evolutionary history also, it becomes systematics, again by Carlos Linnaeus. Father of taxonomy and systematics will be Carlos Linnaeus. Now, you have to remember ICBN, that is International Code for Botanical Nomenclature, and International Code of Zoological Nomenclature. They will help us to name these organisms. All the taxonomical hierarchies in the ascending order, you should know. This is the ascending order that starts with species, genus, family, order, class, phylum, and then kingdom. Okay. Now these terms are given. I'm leaving you with these notes. You must remember this. You must remember this. So Leo, Tigris, and Pardus. This is not cheetah. This is your leopard. So lion, tiger, and leopard, Leo, Tigris, Pardus, they come under one genus, that is Panthera. Cat is Felis. Now both come under the same family, which is Felidae. Then we have dog family, Canidae. Both come under the same order, Carnivora. We have other orders also, and they together make one class, Mammalia. Then Mammalia and many other classes combine to form Chordata. We have read Animal Kingdom. Chordata and all the non-chordates, they form the kingdom Animalia. Are these points clear to all? Please let me know. I'm leaving you with 10 questions, statement-based questions, and more questions will be discussed in the special classes. Now, quickly give me a thumbs up. Let me know, how did the session go? Were you able to revise? Were you able to revise? Doesn't matter, Vinita. See, plant kingdom took us less time because I had already made detailed notes for that, right? So I thought plant kingdom should go in flowcharts. So see, that is the significance of flowcharts. Flowcharts help you to revise things so quickly, right? Thank you so much, everyone. I will see you again tomorrow in the special classes. Time will be informed to you. Also, day after tomorrow, we are going to discuss the second unit, which will be morphology, anatomy, and structure organization, including cockroach and frog. I'll be bringing all these point-wise notes for your revision. Okay. Are these points clear to everyone? So I'll see you day after tomorrow. Thank you so much for joining me. And don't forget to attend. What? Don't forget to attend. Hanji, what have I told you? You have to take that mock test. All of you have to take mock test from 19th of April 2 to 5. Is that clear? Thank you so much. You will be getting these notes. Now revise and get ready for the next class. Bye guys.